Well, good morning, everyone. I hope you enjoyed the wonderful musical accompaniment provided by Sam Dosher, who is one of the ones we'd like to thank today for helping from NAI Central in organizing and structuring this event. So, Sam, that was wonderful. And Mike, I wanted to say thank you, too, for bringing Dunkin' Donuts this morning. So those of us sitting here in Colorado are well sugared and fed to start our day. But more seriously, really thrilled to see a lot of people online and ready to participate today in this workshop without walls. It's focused on serpentinizing systems science. And it's been under discussion over the last year. I'll give a little bit of a history of how to enact such a workshop and try to use this as an effort to build a sense of community amongst the disparate areas of research that are going on in serpentinizing systems science at the moment, both supported by NASA and by multiple agencies, including many of our international partners, some of which could join us today, which is exciting too, from both Europe and from Japan. In the flyers that have been distributed, I think the way it's been described so far is that one of the hopes over these next 3 days that we're going to be convened is that we'll highlight recent advances in understanding how serpentinizing systems function chemically and biologically and geologically within our solar system. And in many ways, the question is, why are we having this workshop now? And it is to come back and re-examine many of the couplings between these three multiple processes and to understand them in the lens of why serpentinizing systems are a compelling astrobiological target in our search for life in the solar system. And it's an interesting discussion that came up about a year ago at AbSciCon 2015. The idea is that serpentinization has been discussed in astrobiological terms for a long time now, and it's extraordinarily well studied as a geological process. And the reason that it seems timely to have a workshop like this and again to be brought together from these various disciplines is to really ask then, what do we understand about the conditions that give rise to an inhabited state in serpentinizing systems? Have we been able to start to recognize when that occurs? What is it, when and where does the hydration of ultramafic rocks actually give rise to states that can sustain life activity? Because not all serpentinizing systems necessarily are alive. When and are they? How do we know that? And I'm hoping we'll explore that again through a bunch of the proposed talks that are in the schedule, as well as in some of the pop-up talks already planned at the moment, and some that might get inspired by the discussions that are going to happen. So Lisa, to give you the next slide. Mm-hmm. This is just was me on my iPhone this morning, going back to June 2015. There was about 40 of us convened in an impromptu discussion at AbSciCon in the middle of that week, just to, again, bring together people in many of the diverse areas that relate to serpentinizing system science to ask the question of whether there needed to be broader discourse about some of our recent findings. And the strong answer from that was yes, and the question has been then how to best do that. You can see the guy sitting in the back right corner there, Tori Holer. He jumped to his feet and said, "We must do this." Yes. Jen Blank, who is also a co-convener of this, jumped to her feet and said, "We must do this." But more seriously, the two of them have been absolutely wonderful in helping to create a framework and a structure for putting together a workshop without walls as an outgrowth of this. In the beginning of the discussion of whether or not there should be the establishment of a working group that can further build on potentially some of the discussions we're going to start in these next few days. We don't necessarily need a working group, and so I urge you to consider that as the workshop proceeds, and to please send your comments and thoughts and ideas about that directly to Jen, and she'll have her email up again later in the workshop, Jen Blank at nasa.gov. to be able to take some of the input and information on that. Because regardless, I think we have a lot that we can do together in these next 3 days, again, trying to highlight some of these recent advances. So if you go to the next slide, that'd be great. Okay, so again, I'd really like to thank the NASA Astrobiology Institute. There's been many discussions through both Executive Council and with NAI Central about ways to support what they could see as a lot of potential synergy in research themes between programs they're directly funding, such as some of the members of the different NASA astrobiology institutes, a lot of active ongoing research in serpentinizing system science in many of the teams at the moment, but also well beyond necessarily any of the specific ongoing institutes, but on other NASA-supported programs and in many research labs, again, internationally. And so how to make sure that the best collaborative outcomes and building upon each other findings could happen. And so, again, to Mike and Estelle and Sam in particular for helping us figure out how to structure a workshop like this, I say thank you. So one of the overarching discussions I think that can occur as we move forward is trying to figure out how to continuously reintegrate some of our perspectives from planetary surface processes, where we see evidence of active serpentinization in our solar system, as well as past serpentinization activity, to any evidence for inhabitation of those systems. And we'll treat a lot of this workshop in the reverse direction. We're going to start with looking at activity in serpentinizing systems on Earth right now, because there's so much active research in that realm, and build it back towards how does this fit into our understandings of planetary surface processes that give rise to serpentinization or are dependent upon it. Again, just trying to think about all of these different research themes that are occurring at the moment. If you'll move to the next slide too, we hope to capture just a fraction of them in the 3 days that are coming. So we're really lucky to have 3 theme leads, Billy Brazelton, Tom McCollum, and Steve Vance. All of them were essentially requested through a lot of that community input, AbSciCon and in follow-up discussions, as the 3 people that most people would like to see bring together perspectives on these topics, which are going to be the focus of the 3 days that come. So in particular, we're going to have a chance to look at serpentinizing systems on Earth, and there will be a very active focus on what we are starting to understand about geochemical dynamics and microbial activity within those systems. There's so much active research at the moment at sites worldwide that the discovery potential has been enormous regarding the types of life activity and how we recognize it, the physiological adaptations to the conditions that exist in actively serpentinizing systems, and almost can't keep up fast enough with hearing who and when and where some of these biological processes are being supported by serpentinization. At the same time, there's experiments being continuously, oh, I'm getting dizzy from Billy. Hey, good. It's interactive. Hey, I can see you guys. So when we look at what's going on in experimental systems, there's been a large number of recent experimental results, particularly in the high temperature realm, again, looking at the pathways of serpentinization and how that process proceeds, and the new insights into some of the products that are occurring, particularly with respect to carbon compounds in serpentinizing systems. But we need this feedback between the state of environments undergoing serpentinization and the experiments that also best mimic that and allow us to see the rates and the mechanisms of these processes and how they feedback. So we have a chance again to look into the structure of new experiments that are being designed, modeling that has shown some of the most fruitful regimes to try and either simulate in experiments or go seek in the natural environment. I think we're going to get a chance too, even in some of the pop-up talks, I hope, to hear about some of the new designs of reactors and fuel cells and microfluidic devices in ways that allow us to really experiment and test how organisms function under the in situ condition of serpentinization and again, some of the boundary states under which that can occur. And then lastly, again, into this realm back into the planetary is going as our advancement in the theory and observation of life activity and habitable conditions in serpentinizing systems expands. How are we feeding that back to our ability to seek and detect those environments elsewhere, particularly in our solar system as well as beyond? How are we, in many ways, both becoming more adept at identifying actively serpentinizing environments right now in the chemical signatures of that, and evidence for where this has occurred in the past? There's a giant open door that has many open questions regarding what are we looking for in those serpentinized environments that might really be an indicator or of a signature of life activity, a biosignature hosted by a serpentinizing system, let alone the taphonomy of those biosignatures and how they're preserved. But what are the true targets that we're starting to recognize that we should be incorporating into our life detection strategies and into our mission targets, both where we're landing or where we are sending orbiters or sending probes, and how we would deploy in that mission to detect life that we believe should be sustained by serpentinization activity. So again, we could have gone in any order with these three, and the hope is that these themes can feed back on each other over these next few days, and we can at least keep these concepts in our mind as we change from the topics that Billy and Tom and Steve are going to lead for us in trying to, again, build a base of recent discovery and state-of-the-art of our thinking in many of these topic areas. So with that, I'm excited to be able to sit back some, but I hope there'll be a lot of active chat in the windows as people are thinking about things. Use these other channels too to inject some of your thoughts as you hear these speakers, and everyone feel free too to comment and reply, because that's where we'll get a lot of the vibrancy of this event. And with that, I can see you, Billy, and we send it off to you. And thank you for leading today's event.
So Alexis, actually, Adrian Brown's asking in the chat room about the background picture of your intro slides.
Yes.
We have a couple minutes if you want to.
Do we? Yeah,
humor him.
Hi, Adrian. I can't see you right now. We had a lovely visit when Adrian came here this summer. That picture, and can you even delete the text off of it? Mike, do you have editing?
No, we can go back.
It's not a big deal. That picture. Yeah, that picture is common to a lot of different serpentinizing systems. That specifically comes from Oman, in an area that we visited with a close collaborator, Peter Kellerman, that we work with. These are rocks that even the more dramatic version of them is strongly banded from red to green, and then has all of this veining across it, with essentially strong oxygen fugacity gradients. Incredibly interesting mineralogy that no one can quite figure out still. So there's some great challenges in what the true mineralogy of some of these serpentinites are. But in this particular case too, there's been an extensive fracturing and evidence of fluid flow and mineralization that has occurred in the system. So there's so many different states of the environment captured in something like that rock that's pictured there, because in the dark green matrix itself is the area where all the reactions are occurring that, for example, may be producing molecular hydrogen or reduced organic compounds. But that fluid environment of the high transfer potential, very different, again, redox state and oxygen fugacity, may be, again, an area where there is a lot of mixing and dynamics of fluids from different parts of the system, where we may find, say, an enhanced habitable condition and evidence of biosignatures. And understanding how to seek and unravel those kinds of rocks is definitely a great challenge at the moment. I do want to say, too, for those of you who are thinking about pop-up talks and things, some of the pop-up talks might be a few slides, but if you just want to inject one image or one idea, again, please feel free, not for today, but to send your requests forward for the next 2 days to put those ideas in. And again, you get inspired. At the moment, there'll be one student from here at Colorado, Katie Rumford, who's going to do a quick pop-up talk today. I see Hannah Miller here as an example, and I would have had requested that she give a pop-up, except that she defended her PhD thesis yesterday, and I was trying to be gentle and kind after such an important event. But the reason that I would have asked, for example, someone like Hannah to give a quick pop-up, and you might want to encourage your students and postdocs and other research scientists working with you to give a quick pop-up, is two major outcomes from her thesis work, which are starting to come out into various publications, are one, some of the key reactions that actually make molecular hydrogen at temperatures within the temperature limits of life. So she's been trying to see reactions within actively serpentinizing rocks that make hydrogen and identify those, and that's been really changed our thinking about where to go look for life activity. The second one was a part of her thesis that worked on looking at Methanogens, a type of life that we think should be functionally active in serpentinizing systems, who they are and how they work at the high alkaline pHs that we often see in serpentinizing systems, and the carbon isotope signatures of the methane they make. So depending on how things evolve, I may have to twist your arm and ask you to talk about that later this week. But for now, again, sending it back to you, Billy. Thank you.
Thank you, Alexis. Yes, as Alexis said, I am the day one theme lead, which is serpentinizing systems on Earth. So we have the very specific topic of only serpentinization on Earth, not other planets. You see the schedule here. Before we get to our 4 featured invited talks, I'm going to give a quick scientific overview of serpentinization. So go ahead and go to the next slide to give just some background information, because we want to be welcoming to everyone, even those who may not already be familiar with serpentinization. This workshop is not intended to be exclusive to serpentinization fanatics. We're welcoming everybody. So here's an introduction for those people. We know of 2 energy sources in the solar system that we know can support life. There's the sun, of course, that supports all green plants and therefore all other surface-based life on this planet. Everything you eat comes from the sun. And then when hydrothermal vents were discovered in the late 70s, we realized that magmatic heat from the Earth's mantle can also support ecosystems on Earth independent from the sun. So from an astrobiological perspective, when we're looking for life, we need to find solar-powered life or geologically active planets, or both. With the recent discovery of serpentinization, I can control the slides. Yes? No? Next slide, please.
Yeah, Billy, click the down arrow in the lower left of the slides. Do you see
The down arrow, not the right arrow.
Okay.
So here is my very simplistic introduction to serpentinization, and later on in this workshop, I'm sure we will get more comprehensive and more precise definitions of what serpentinization reactions are and what they can do. But my microbiologist's definition of serpentinization is here. It's the process by which the pretty green rock turns into the ugly black rock. And from a biological perspective, what's exciting about that is that it's exothermic, it releases energy, it releases hydrogen, the hydrogen can potentially create organic matter that can support life. So this is potentially a 3rd source of energy that could support life. This is one of the things that everyone, many of the people involved in this workshop want to be able to study, is can this support life, and can it in fact support life independent of the other 2 sources of energy we know about, stars and magma. Is serpentinization or geochemical reactions a 3rd kind of energy that can support life on their own? We don't know. That's what we're here to discuss. And to make this a little bit more concrete in my short overview, I'm going to give you one example of a serpentinizing system on Earth. This is the Tablelands Ophilite in Grossmoor National Park in Newfoundland, where Matt Schrank and I have been working with Penny Morrill, who's at Memorial University in St. John's. And I really like this picture because it's an overview of the watershed where we find pH 12 springs coming out of serpentinites. And I also like this picture because it looks a little bit like Mars, which is not entirely a coincidence. The serpentinites are toxic to most plants, and so the vegetation is very sparse. Here's some beautiful pictures of serpentinites. You see the snakeskin texture, which gives the name to serpentine, serpentinite. And here, I think I have to do this. Cursor. Now you can see my pointer, I think. Nope. Okay, how that works. Right around here is a spring coming out of the serpentinites. And what I want to show you is that this show you this as an example of a conceptual and intellectual challenge we have almost anywhere we're going to study serpentinization. So in this site, you have subsurface water coming out. It's very high pH, very extreme, and you have surface water, in this case coming from a swamp that is flowing into this pool. And there's a mixing zone in between them, and we look at the microbial diversity at these 3 spots. We see a lot of kinds of microbes coming from, I don't know how that works.
Billy, if you just click where you want the pointer to go, it's better than dragging it.
No dragging, just click. Got it, thank you. Lots of different kinds of microbes come from the swamp, as you might expect. Very few kinds of microbes are coming from the subsurface source. Okay. And where they mix, you have a mix of microbes. That is not surprising. So our challenge when we study places like this is that what we really want to know is what is going on in the subsurface where it's not affected by the oxygenated Earth's atmosphere, right? We don't really care what lives in the swamp. If we were a swamp biologist, we would care, but we are studying serpentinization and we want to remove that effect of the microbes coming from the swamp. And we're also not that interested in the mixing zone, although it's really biologically interesting. That's usually where we see the most biological activity, because mixing zones are where biology really thrives. And one of the big questions that I think will be a theme of this workshop, especially day one, is how can we figure out what's going on in this heavily serpentinization influenced environment, even though we have to work on the surface of the Earth and it's contaminated and mixed with all these other things that we may not care about. And here is one more slide showing that this is a general trend. This is a data set I compiled several years ago. It's old now, I need to make a new one, but it very nicely illustrates this point that as pH goes up, and in places where I got this data, pH is an indicator of the degree of serpentinization. As the pH goes up, as serpentinization becomes more and more important, the diversity, and the units on the y-axis are not so important here. What's important is that the diversity of the microbes is going down as the pH goes up, as serpentinization becomes more important. And in many of the places, not only is the diversity low, the biomass is also very low, as an indication that these are very difficult places to live. So this magnifies the problem of contamination of the surface, because the surface has more stuff, it has more kinds of stuff, and it totally swamps out the signal from the subsurface in many cases. And this is a very challenging problem that we will also, for example, probably have to deal with on Mars or any other planet that we study. And later this morning, you will hear from Matt Schrank more about the microbial diversity of microbes. That's just my quick overview for now. One more point is that example I just showed you was one type of a serpentinization system on land. Here's another one in Italy. You can also have serpentinizing systems on the seafloor. This is the Lost City hydrothermal field and these huge carbonate chimneys that form on the seafloor. And the point I want to make is in both cases, all of the biological activity that we can easily study anyway is occurring at these mixing zones. So I showed you the example at the tablelands, where most activity is where the subsurface water comes to the surface and mixes. That's most of the activity that we can detect. Lost City is the same thing. These chimneys are formed by the mixing of subsurface fluids with surface fluids. The chimneys are only there because mixing is occurring. And when we have visited Lost City in the past, we have mostly studied these beautiful carbonate chimneys with tons and tons of life on them because they were the obvious thing to study. But if we want to have a general understanding of how serpentinization and life interact and sort of universal fundamental principles, the chimneys may not be the best place to study. We may want to study things down here or over here. And we just haven't done that yet in many places, although we hear about some new studies later today. So this is just a textual summary of what I just said. Chimneys are mixing. This is what we've studied before. It's probably not what we want to focus on in the future. And once again, the reason why we need to be able to sort out these subsurface versus surface processes is that's probably how we're going to be able to address the question of whether serpentinization can support life on another planet, for example, where there may not be an ocean of oxidized seawater above it, or may not be sunlight providing oxygen and things like that. So this is one framework for understanding what the invited talks are going to talk about today. What is going on underneath the chimneys? What is going on in areas where there's less focused flow, but more widespread on a spatial and volume basis? And so these are what among the topics that the talks later today will talk about. So next up after me will be Susan Lang, who will talk about the carbon chemistry of serpentinizing systems like this. After Susan, Everett will talk about the energetics of these molecules and how these chemical reactions may or may not be useful to biology. And then Beth will talk about a recent drilling expedition where we drilled into the rocks around the Lost City hydrothermal field directly into the serpentinites to really get a more direct study of life in the subsurface that may not be quite as affected by the surface. Although, of course, that is one thing to be determined. So I think you guys get my points here, but here's one more slide to drive it home. On Earth, we have this challenge that we mostly study things on the surface, but what we really want to know is whether there's some green slime down in the subsurface that's actually doing the astrobiologically interesting stuff. And if we send robots to Mars or to Europa or to Enceladus, I think that we will have a very similar intellectual problem on our hands, which is that we mostly measure things on the surface, but what we really care about is if there's some green slime in the subsurface that's really doing the interesting stuff. So I encourage you to think about this kind of conceptual theme as we listen to the invited talks and to the pop-up talks at the end of the day. And I think it might be a useful framework for interpreting the data that will be presented. So that's all I wanted to say. Yeah.
Before you go ahead then and start with all of the talks that you lined up, we already had some commentary in the chat, and Darcy Meyer-Dombard, and Darcy, I do want to say as well, was someone who's been very willing to help spearhead the idea of a working group, and I'd love to hear your input on that after this workshop too. She says, just a comment on the concept of the mixing zone and interest in them. If the mixing zone is a true ecotone, we can sometimes see cool examples of the subsurface influencing what is going on in surface-affected environments. And it just seemed worth having you comment on that a little bit more before we go into the next talk.
Yeah, that's a really good point. I perhaps presented it as mostly a one-way interaction where the surface is swamping or obscuring the interesting stuff in the subsurface, but it's definitely can be a two-way interaction where the subsurface is influencing the surface, and that's where a lot of interesting science is going on, and perhaps even by necessity, that's the only way that we can actually understand what is going on in the subsurface is by understanding how it affects the surface, and then inferring what the subsurface without the surface influence might have looked like. So yes, I think that's a really good point. Yeah, so that's another way to put my recommendation to our participants is when we see data from field systems or perhaps even experimental systems, it's good to think about, is this a surface process swamping out the subsurface? Is it subsurface actually emerging despite the noise from the surface, or some interesting synergy of the two? I think that might be one way to think about these different kinds of data we'll hear about soon. So you hear the schedule here. We have a full slate of pop-up talks at the end, so I am really looking forward to that. And I want to encourage everyone to ask questions. Probably limit the questions out loud to after talks, but certainly if there's an important permanent question during the talk, I think that may be possible as well, but certainly after talks, and especially anytime during the talks, in between talks, in the chat window. We are hoping the chat window will be a really vigorous field of intellectual discourse. The whole purpose of this is to stimulate lots of really good discussions.
Hi, Billy. We also had a question from Adrian Brown asking if you could comment on the mineralogy you're seeing at the surface where the spring and the surface waters were mixing. So that probably, I think you meant the white mineralogy.
They are serpentinites, and I know that serpentinites are rich in serpentine, and that is my extensive geochemical knowledge of the mineralogy of those rocks. Someone else could probably better answer that question. But they are really beautiful serpentinites. I can vouch for that.
Thank you.
Okay, I will let Susan take over.
Hi, thanks, Billy. Okay, it looks like my slides are up, and thanks for the opportunity to chat today. So Billy had asked me to talk a little bit about carbon and serpentinization environments, and asked me as well to talk as much as I could comparing and contrasting some of the signatures that we see in oceanic versus continental systems. I'll do that to the extent that I can, but the focus is probably going to be a little bit more on oceanic systems. And so the image that you see on the left is from the Lost City hydrothermal field, and then the one on the right is from a continental system in Liguria. And in both cases, you can see these large carbonate deposits that kind of typify some of these serpentinization systems. And so, as Billy introduced already, one of the outcomes of the serpentinization reaction are these hydrogen concentrations, and Alexis mentioned this as well. And part of the reason why I and many other people are very interested in that is because that hydrogen can go on to fuel both the abiotic synthesis of organic compounds, and it can fuel microbial growth, autotrophic growth in the subsurface or as those fluids move to the surface. And when we think about those processes, most of the time the reactions we're thinking about or the metabolisms we're thinking about require something like inorganic carbon as a starting material. A lot of times we kind of default to thinking of it as dissolved inorganic carbon, so something like bicarbonate or CO2, but in general it's oxidized carbon. Those processes, both of them will alter carbon signatures both in fluids and in rocks. And so as a result of this, some of the research I'm interested in and what I wanted to talk a little bit about today is what happens to carbon in general in serpentinite-hosted environments. And the way I thought I would organize this is by, those looked a little bit more like arrows on my on my version, but I think you get the picture that the organization I wanted to try and go through is going through this as a fluid flow pathway. And so in the ocean, you have seawater that downwells. In the subsurface, there's some sort of mantle input that we can see geochemical signatures of. There's the possibility of picking up material from things like fluid inclusions, so small bubbles in the rocks that can include things like methane. And then as those fluids are heated, they start to buoyantly rise and they exit out the seafloor in these large chimney structures. You can have similar processes that go on in the continents, and people in continental systems have pointed out that in these environments you can have two different types of fluid pathways. First of all, you're starting with something like meteoric water, and if that water as it passes through the rock is in connection with the atmosphere, and you're continuing to have carbon dioxide input and oxidants from the atmosphere, it's an open system and you have one type of fluid chemistry. And if you've sealed that fluid pathway off from interaction with the surface, then it's a closed system and you have a very different type of fluid pathway. And again, you can have the potential for something like fluid inclusions in the subsurface. And so one of the first things to think about is what your fluid input is and what happens to that material as it passes through the system. And so in oceanic systems, you have a pretty homogeneous starting material of ocean deep seawater where you have dissolved inorganic carbon present at about 2 millimolar and seawater dissolved organic carbon present as about 30 to 45 micromole of organic carbon. In continental systems, this is a number I picked up from Oman, and in open systems, you have about 1 to 3 millimole of inorganic carbon that's present in those fluids, and you also have meteoric inorganic carbon and dissolved organic carbon, and the amount that's there can probably be fairly dependent on whether or not the fluids are going through something like a soil layer. And so one of the first things to think about is what happens in particular to that inorganic carbon. And this is a picture of a serpentinite that was recovered from the Atlantis Massif. And similar to the pictures that Billy showed, there's this fine veins of calcium carbonate material And so we know from a lot of work from a lot of people that a lot of that inorganic carbon that's brought in with fluids is precipitated out as calcium carbonate. And so just to highlight two examples, one is here from the Iberian margin, where Esther Schwarzenbach and coworkers looked at the amount of total inorganic carbon that was present in rocks. And where you have cold shearing and you're starting to have fluids pass through those rocks, you have a lot more inorganic carbon that's precipitated. And then a recent example from Oman, where they were looking at travertine veins and the C14 ages of those travertine veins, where again you have a lot of inorganic carbon that's been precipitated in these veins over a pretty wide range of time frames. And so one of the questions that's still being constrained is how quantitative and how rapid is this calcium carbonate precipitation? And the answer to that will have an impact on both what material is available as a starting material for abiotic reactions, for microbes in the subsurface, and obviously a lot of people are interested in it from the standpoint of trying to quantify these systems as a carbon sink. And so once the fluids are in the subsurface and are in some sort of reaction zone, it's a little hard to start distinguishing what happens in that area, in that region, versus what's happening in the surface, which is something that Billy also alluded to. And so what I thought I would do is now focus a little bit more on oceanic systems and look at some of the end member fluids from one particular oceanic system and try and use some evidence to talk about what might be happening in the subsurface versus what's happening more towards the surface. But really the easiest way to go about that at this point is to look at some of the fluid chemistry in the end member fluids, although folks will be talking a little bit later today about some other approaches to try and get at what's happening in the rocks versus what's happening more towards the surface. And so the system that I'm going to focus on is the Lost City hydrothermal system. The map in the upper left is the location of the field, is there in a white dot. And on the bottom left, there's a cartoon of the way we visualize what might be happening with the fluids. And in general, what we think is happening is that there's a single end member fluid that's feeding the field, and towards the center of the field, you have higher temperatures and higher hydrogen concentrations. And then as you move to the periphery of the field, fluids are conductively cooled. They continue to undergo water-rock reactions, and you have a drawdown of hydrogen, we think mainly because of sulfate reduction. And so these fluids that are feeding the chimneys on the edges of the field, we think have been more exposed to microbial activity. I've tried to list here all the carbon species that we've identified to date in the Lost City fluids. I think it's pretty comprehensive, but I could be wrong. And I've organized it in by oxidation state. So I've already mentioned that there's a lot of calcium carbonate precipitation. So CO2 in the surface is precipitated out as calcium carbonate, and so not surprising is that these end member fluids have very low inorganic carbon present in the fluids. And then on the other end of this list is very reduced material, so things like methane, and there's high concentrations of methane, but that can be challenging to use as a starting material in the anaerobic end member for either abiotic reactions or for microbial metabolism because it's already so reduced. And so the question as we're looking at what is present in these fluids, so methane is the most abundant carbon species that's present in the fluid, followed by the single carbon organic acid formate, which is present from 34 to 140 micromole. And then you have things like amino acids and acetate that are also present. And so one of the questions that we've been trying to address is of these, what's biologically produced and what's abiologically produced. And there's a couple ways that we go about that. We can use geochemical evidence to try and address that question. So with things like amino acids, we can look at the distribution of the amino acids, compare them to what we would expect a biological signature to look like. So what's the distribution of amino acids in the surface ocean? What's the distribution of amino acids in an autotrophic culture versus a culture that's been fed, that's heterotrophic and has been fed phytoplankton or something along those lines. And so there's just pure distribution arguments that you can make to try and address some of these questions. And from that, we have indications that amino acids, for example, are biologically produced, and things like acetate is also most likely biologically produced, and that's coming mainly from the signatures of where acetate is highest, and we see the most acetate where we see evidence of the most microbial activity. So it's most likely something like a microbial byproduct. Another way we try and address these questions is using C13 data and since people have done some very extensive testing in benchtop experiments to try and look at fractionation factors, we can start trying to use C13 to create an isotope budget and look at sources of carbon and synthesis pathways. And so that's also been a very valuable tool. And then the final way, or one of the other ways that we are trying to approach these questions, is by looking at C14 signatures. And so that's what I wanted to talk a little bit more about. And so this is a graph of C14 signatures goes from about 0, and that's where Mantle input comes from. So Mantle carbon is C14 free. And on the other end of the spectrum, you have modern seawater, which has a C14 signature of approximately 1. And so from some C14 work is very useful in this type of environment where you have these very large end members, very large differences between the end members. And so you can start to distinguish at least where the carbon sources are coming from, and from that try and infer what the process is. And so based on some previous work, the local seawater dissolved inorganic carbon, not surprisingly, is modern. The chimneys, as Billy mentioned, come from this mixing zone, this mixing of this high pH fluid with local bicarbonate. And so those carbonate chimneys are also modern. And then the methane in the system is C14 free and has a C13 signature. These numbers on the bottom of these is the C13 signature. And so that methane has a C13 signature of about -12 per mil and is C14 free. And I thought I would mention that, and so let me first say that, so in Lost City, it's thought that the methane is largely abiotically produced, although there's methanogens present who are probably making methane. And so there is probably a biological component to the methane as well. And there's still questions of whether that methane is created kind of on the fly as the fluids pass through, or if it's being harvested from fluid inclusions. And I did want to mention, just going back to continental systems, that there's methane present in those fluids as well. I've seen a couple of numbers of C14 from methane, both in the literature and from work that we've done in the lab, that methane is C14 free. And so again, you need to have a source, initial carbon source from that methane of either coming from some C14 free material like calcium carbonate that's been down there for a very long time, or from fluid inclusions. And so I've also plotted on here the formate, which is data that we generated recently. And so depending on what location you are in the field, we have these different chimneys, and you have different concentrations of formate across the field. And so depending on where you are, this formate is largely C14 free. And in other places where we have other kind of indications that there's some local mixing with local seawater. Some of that formate is starting to include a relatively modern seawater signature. And so while we were interpreting this, the interpretation that we've settled on is that the C14 free formate has to be coming from the subsurface, where you still have a mantle CO2 signature that's available, whereas this additional input of more modern formate is most likely biologically derived, because this is happening in temperature regimes where microbial activity is a lot more favorable. And so if I was just going to be extraordinarily simplistic and just put some check marks on things, I would say that something like methane is predominantly abiotic, and I haven't talked at all about the short chain hydrocarbons, but based on some geochemical and some C13 signatures, there's also indications that that's abiological, whereas amino acids and acetate are most likely biological, and formate probably has this mixed signature. And so now as we're moving up and definitively in the chimneys, there's been quite some work in those environments to, first of all, we know there's a lot more carbonate precipitation because there's large carbonate chimneys, but there's been quite some work by people like Matt Schrank and Billy Brazelton to characterize the communities that are in those environments. And there's a fairly significant difference between the communities that are inhabiting the interior of these chimneys, where it's dominated by anaerobic processes, and the exterior of these chimneys where you see more exposure to local oxidized seawater. And so, and this is just a picture, an up-close picture of some of the dense biofilms that you can see in some of these chimneys, and their very close association with things like brucites. And so one approach to try and get a better idea of what's happening with these microbial communities in these chimneys is to look at things like specific biomarkers that these communities can form. And so if you are talking about bacterial communities, a lot of these bacterial communities produce fatty acids in particular, and from some C13 work of Alex Bradley and coworkers, they've already shown that in the exterior chimney, or they've interpreted that the C16 fatty acids with a single double bond in them have a C13 signature of about -20, and they've interpreted that as indicating that there's an exterior, that it's coming from one of these exterior microbial communities that has a little bit easier access to local DIC. Whereas in contrast, as you get into the interior of the chimney, the bacteria don't have as easy of an access to inorganic carbon, and the C13 signatures turn to be more around -9. And then the other community that's present in the interior of the chimneys is the methanosarcinales. The lost city methanosarcinales in particular tends to dominate the anoxic interior of a lot of these chimneys, and they produce a very diagnostic biomarker called PMIs. And so these numbers that are up here right now are actually the C13 signatures of one particular chimney in the lost city field that we took, and we analyzed the C13 signatures of them, and the numbers are consistent with what other people have seen in the past. And then we went on to also look at their C14 signatures to try and get a little bit more insight into what their metabolisms were. And so consistent with what was suggested just based on the C13 data, the bacterial community is kind of split. And so the biomarkers that have a more negative C13 signature have a more modern signature, and whereas those that seem to be undergoing some sort of carbon limitation have a more C14 free signature. And whereas the methanosarcinales and the PMIs have this more mixed signature. And some work that Billy Brazelton has done has demonstrated that it's very likely that these archaea may not be able to take up formate directly, and so it may be that they're relying on some sort of inorganic carbon source. And so the way we've interpreted all this is that you have some sort of formate source that's coming from the subsurface and abiotically produced. It's utilized by sulfate reducers, and in the formate dehydrogenase gene, you have rapid conversion between formate and inorganic carbon, and then that inorganic carbon can mix with local seawater inorganic carbon, and this new mixed inorganic carbon pool can be used by autotrophs, or these sulfate reducers could also potentially produce organics that are used by heterotrophs. And so just to wrap up some of the major things that we're thinking about, so during downwelling, you have the removal of inorganic carbon, the question as to the fate of organic carbon that's present in deep seawater, and hopefully Beth and Matt will talk a little bit about the biosphere in kind of these early rocky locations. In the subsurface, you have this mantle input of inorganic carbon, probably conversion to formate. There's also evidence that there's precipitation of this mantle CO2 as bicarbonate. You can see that in some of the 18O data from rocks. And there's conversion, or there's addition of methane and alkanes. And then there's the seafloor expression, further precipitation, and microbial uptake. And so I will leave it there and am happy to take any questions. I can see there's questions over here.
Yeah, thanks, Susan.
Acetylene detected. Not that I know of, but I don't know that. I think it would show up in some of the analyses that we've done, particularly the short chain and alkane analyses. We should see acetylene, and so I don't think that anyone's seen it, or at least I've not seen any reports of it. And the C13 might be misleading if examined in other settings. I would say the C13 can be misleading if examined in this setting. And so, yes, I think C13 is great. Sorry, I will reread the question. C13 might be misleading if examined in other settings. Have similar trends been observed in other vent systems? Do fatty acids have diagnostic oxygen and hydrogen isotopic signatures? So again, I would say C13 can be misleading in this setting. The great thing about C13 is it records, in addition to recording the source of carbon, it's recording a process that's happened to that carbon. But you have to consider that the signature of the source can always change based on other processes that's happened with that source, and that the signature of the process can change based on things like temperature, surprisingly, things like how much energy is available, so how much hydrogen is present can change the isotope signatures of the newly created material. And so C13 has to be thought about very, very carefully. The fatty acids look the delta D signatures of the fatty acids look pretty similar to what you would expect for microbial activity. And so if the question is whether or not you can use that to distinguish between biological and abiological synthesis reactions, I'd have to think about it a little bit more. The fatty acids look biological, but I can't think off the top of my head of what the DH signature is of alkanes that have been produced abiotically. So I don't know that off the top of my head.
Susan, this is Alexis. I just want to stop you for a sec because we're getting so much information. No, it's fantastic, and I'm just sort of trying to absorb it. And I think Billy has a bunch of questions coming in from the audience that he can walk through with you, just so we all have time to see them separated out a little bit. It'd be great with the other speakers too, if we can have a look, take that down, so we have just a little time to know what the questions are too. Thanks.
Thanks for addressing my question.
So how do you, so Billy, do you want me to, how would you like to proceed?
Billy, I think you're muted. I think you're trying to speak.
Yeah, sorry. I'm not sure either. I guess we can move on to Amy's question, and the first half of it I can answer for you, which is that we don't find any obvious acetogens, but acetogens are not always obviously easy to detect. And then I'll let you answer her second question, which is, do you think this is being formed in the downwelling location?
I think that the acetate is being, I assume you're asking if the acetate's being formed in the downwelling location. I think it's being formed in the chimneys based on the C13 signatures that we have of the acetate. And so they're a little unusual given what you normally see with acetate. And the easiest way to explain them is that the biomass, if you just take bulk biomass in the chimney, it's more enriched in C13 than what you normally expect with bulk biomass, again, because you have such a carbon-limited system. But if you take kind of very traditional values of the fractionation between bulk biomass and the breakdown of that bulk biomass under anaerobic conditions, you would produce acetate. That'd be one of the expected outcomes, and that acetate has a C13 signature that's consistent with that being the process, that you basically have some heterotroph eating up the bulk biomass and producing acetate.
And then one more question from Jen, and then we'll move on to the next talk, perhaps, about whether the C13 changes with the age of the carbonate.
That's a great question. Let me think about that for a second. I don't think so, but I could be wrong. I think that there is. I don't remember the C13 signatures versus C14 signatures off the top of my head. There is a relationship now that I'm thinking about it, and you end up getting more negative C13 signatures where the carbonate is a little bit older, and so. Yes. Assuming that C14 is purely reflecting an age.
A yes/no answer. Awesome. There's maybe probably going to be some more questions. Probably going to be some more questions for Susan, and I'll let her and the rest of us answer them in the chat window, and we'll move on to the next talk. Thank you very much, Susan.
Thanks.
Okay, Everett, I promoted you to a presenter. If you want to start your webcam and then unmute your phone, we're ready for you.
Okay, I think I've done those things. Can you hear me?
Yes, we can hear you. We can't see you yet, though, if you want to start that. You might not have clicked Start Sharing on your webcam.
Well, I can see me, which is kind of disturbing. But
So in the middle of your webcam preview, there's a button that says Start Sharing. Can you click that?
Tell me again where it is.
It should be right in the middle of your webcam view, where you're seeing yourself.
Oh, sure enough. Start sharing.
There you go.
Right, there you
Okay,
have it.
take it away.
All right, well, we're going to crawl out of the ocean and onto the land. And so this image we're starting with is from Oman, where the mountain range in the back behind me there is, a big hunk of the Mantle. And it's an exciting place to visit because you can walk around on the Mantle. It's been studied for decades by geologists and petrologists and mantle dynamics people. And really, probably the Samael Ophilite in Oman is largely what we see in the sort of textbook images of what the Earth's lithosphere is like. The lithosphere being the rigid part of the crust and upper mantle of the Earth that is the parts that are actually getting pushed around and as plates on the surface of the Earth. Now, let's see. somewhere here. I'm sure I'm supposed to see an arrow. There it is! Fantastic. All right. So just quickly to orient ourselves, Oman in the southeastern corner of the Arabian Peninsula. You can see, let's see, where is that arrow thing? Ah, there it is. You can see that there are all these mountains on even this map. And if we look at a satellite image, then this mountain range here is these are the ultramafic rocks that are the dark rocks here that come from the Earth's upper mantle. As I said, this has been studied for decades by geologists, and the general story is summarized here let me move that arrow out of the way, in a nice cartoon view. You have mantle peridotite, ocean crust, and then thrust faulting that managed to slice down into the mantle peridotite and send that material up onto the continent, which is, rather unusual. It misbehaved and didn't go down the subduction zone like would be expected. So the fabulous thing is that it's exposed at the surface. You can walk around and have a view, eat lunch on the mantle, various things like that. It's pretty spectacular. Here's a zooming in on the area. The point here is not so much the details, but that these various locations are places where water appears at the surface. It's an arid climate, as you could tell from the satellite image and the first picture. But there is an active hydrologic system, and when the water that infiltrates and reacts with these ultramafic rocks returns to the surface, it has this fantastic properties of very high pH, pushing up towards 12 in what we've measured, and very reduced such that it contains abundant dissolved methane and hydrogen. So those are the rather. Okay. And the point being that has been made at the beginning of this meeting that, it seems rather rare to find serpentinization seems rare at the surface because you have to have these ultramafic rocks exposed, especially on the surface of the continents, much more common on the seafloor. But it's probably extraordinarily ordinary kind of process in our solar system, because this sort of rock type, which after all makes up the majority of the Earth's upper mantle, is probably very common. And in a simplistic way, it's sort of similar to a lot of meteorite compositions. So the process of water reacting with this type of material leading to the alteration is called serpentinization. So I'll move this pointer around a little bit. This dark stuff is the less altered peridotite ultramafic rock from the mantle, and this more the stuff in the veins is the serpentine and carbonate minerals that we've been talking about. In thin section, you see something like this. This is a transmitted light image, and I'll click to a polarized light image. Well, maybe I'll go back there. So you see these large open spaces here, and then these veins cross-cutting in this incredibly complicated sort of system. If we go to the polarized light, you can see that these are large. Let me move the arrow down here. This is like a large grain of olivine here, crossed by these veinlets of serpentine. And then if we zoom in, we see here are these veinlets of serpentine here and so forth. And this stuff in the background then being the unaltered, or certainly a lot less altered rock from the mantle. And then if we look in reflected light, in that same image, you can see these little shiny bits here, these white specks, and those are the bits, in this case, of magnetite reflecting light back to the camera. So you have a sequence of reactions, and we can delve into this in whatever detail anyone wants to in terms of the overall process. You don't always have to have magnetite. You can still have hydrogen production. The connection here is that the iron is being oxidized water is being reduced to hydrogen. Magnetite is just a screaming example of that oxidized iron, going from a mixture of ferrous plus 2 iron, or just plus 2 iron in the olivine to a mixture of ferrous and ferric plus 2 and plus 3 in the magnetite. But you can have ferric iron in the serpentine and elsewhere, and so that creates lots of opportunities for hydrogen production during the entire process. All right, so if we viewed this from petrology, let me move this arrow again. We have olivine and pyroxene, the rocks, the minerals in the unaltered rocks reacting with water and carbon dioxide, and we get serpentine with maybe some brucite, magnetite, carbonate minerals. When we look at this from a geochemical process, we have the olivine and pyroxene reacting with surface waters and air, giving us serpentine, brucite, magnetite, carbonate, and these hyperalkaline solutions that feature reduced gases. And of course, the question that we're also asking is how much of this whole process is populated, where, and by whom, and those kind of questions are being addressed by other people today in more detail than me. So I'm going to focus in here on what can happen. Here is a very simplified cross-sectional view of, again, a continentally exposed ultramafic rocks. Whoa, there we go. Nearly lost my computer. All right, so you have, let me get the arrow. Hey, it's raining. Shallow groundwater, surface water tends to be this magnesium bicarbonate enriched composition. And the deeper water, water that circulates more deeply and hydrates the minerals more extensively, reappears at the surface as basically a calcium hydroxide solution with a very high pH, and this is where the and picks up the hydrogen and brings the hydrogen and methane to the surface as well. So here's an example of a stream system with springs in it in Oman. So this clearer greenish water up here is that surface water, and right around in here, suddenly everything turns white, as you can see. And what's happening is that the calcium hydroxide solution is oozing in here into this stream, and the reactions are happening very rapidly. We have bicarbonate back here in the water, and you have calcium coming in here, and suddenly you get calcium carbonate precipitation. It's all that white stuff. You can even see bits of it back here where there's a little bit of seep coming in under the fresher water. So this is an example of seeing both of those systems. Here we are looking down. There is the toe of my boot for scale. Here's that calcium hydroxide solution. It's coming out of that crack right there, flowing down along this path, falls off the edge of the rock into the stream, and instantly there's a bunch of calcium carbonate precipitation. So this is a telltale sign that we've got the high pH fluid interacting with the surface fluid. And then looking on downstream, it's pretty dramatic in many cases where there's a lot of calcium carbonate precipitation that sets up these kind of stunning views, at least in Oman. All right, so here's a plot of magnesium as a function of pH. And you'll notice that at the very high pH over here, there's very little magnesium dissolved in the water. Those spots that are clustered along the zero axis there are, indeed, there are values associated with those, but this is a linear scale over here, so it's very hard to see those. And then the fresher water, the surface water, is represented by these things over here, pH starting around 8 here, and considerably more enriched in magnesium. And in between, in this zone, is where you would have mixing between the two end members of the fresh water over here, the serpentinized fluid over there, and lo and behold, you get things plotting in between. So the serpentinization process then is removing the magnesium. It removes silica, sulfate, dissolved inorganic carbon. The plots look more or less like this, or these other constituents of the system. Contrast, here's a plot of hydrogen. So here over this entire range, notice there's an entire range, but these high pH, these are the serpentinized fluids, can be enormously enriched in hydrogen relative to these surface fluids. Again, these are measurements, but they're way down in the nanomole level of dissolved hydrogen. It's just still kind of remarkable that even this fresh water has this hydrogen wafting up through it from below. Now you can see that although the serpentinized fluid can be massively enriched in hydrogen, it can also have very low hydrogen, and this is probably evidence of biological consumption of that hydrogen that's being delivered. A plot of methane and a plot of ammonia or sulfide look very much like this, and also calcium, which of course is not directly involved in the oxidation-reduction reaction, but it's an indication of why the pH is going up and what's happening in the non-redox part of the system. There's calcium in the pyroxenes that weathers into solution, and then when the new minerals form, the serpentine minerals, calcium does not fit into those serpentine minerals. It accumulates in solution, and of course you can't just add charge to the solution without balancing it, and so the way that charge is balanced is by an increase in hydroxide. So you end up with a calcium hydroxide solution because there aren't any other anions in the system. The typical anion, something like bicarbonate, has been consumed on the way in as the water is infiltrated in by the precipitation of calcium carbonate. All right. Well, here's a list that we've made of I'm calling here the demonstrated chemolithotrophic reactions in serpentinizing system. And you notice I have these different classes. In fact, there's even something that's fallen off the bottom of the picture. That's pretty good. Anyway, so I've ranked these here with Peter Canovas in terms of, the quality of the information, the molecular and microbiological information. So the best case we have for chemolithotrophic reaction is hydrogen oxidation, and that's because there's at least an isolate and culture of known physiology and genomic data that goes with that, and so we're pretty sure we know something about this reaction. What I'm trying to do here is use what's known coming from molecular biology and microbiology to identify what kinds of reactions are supporting life. What we're calling class 2 type data is where there's something like rate data on redox reaction and isotopically labeled experiments that really show that the reaction seems to be happening and is microbially mediated. And here we have several reactions, carbon monoxide oxidation, the methanogenesis reaction, so making methane from CO2, and there's that hydrogen coming from the, if I point at it, it would work even better. There's that hydrogen coming from the water-rock reaction, and then various anaerobic methanotrophy, simply because often in the literature it's not specified what the oxidant is, and so we're using the three that we have measured here: sulfate, nitrate, and nitrite. Somewhat useful, or pretty useful information, and it's what we're calling class 3 information, where you actually have gene expression. It's not, or transcriptomic or proteomic or biomarker data that indicates the reaction is happening. It's not like watching the reaction happen, but it's very suggestive of that kind of reaction happening. And what fits in here is ammonia oxidation, sulfate reduction. We also have reactions that can be identified based on genes that have been actual gene hunting, looking for genes involved in these sorts of processes. That adds sulfide oxidation and nitrogen reduction to the list. And then what you can't read down here are what we consider the type of information that is least indicative of something actually happening, which is what's inferred from 16S ribosomal RNA data, which is about like saying your brother is a lawyer, so you must be a lawyer. Great. Helpfully, perhaps those reactions are sliced off the bottom of the image. We can't even see them anyway. So there you have it. But there's a couple more reactions on this list, and they'll show up here in just a moment. So one thing we can do with all of this is try to figure out how much energy are we talking about. These are reactions that are identified, have been identified through microbiology or molecular biology, and so what's the energy supply? And so there must be some energy that supports these things. We do this by calculating this term called the chemical affinity right here. And the way you do that is combining, of course, there's the ideal gas constant and temperature, but the important parts over here are the equilibrium constant and the reaction quotient, or the activity product. And essentially, the equilibrium constant tells you where equilibrium is for the reaction, and the activity product or reaction quotient, it's kind of hard to point at, that Q there is what you actually measure out in the field and in samples that you bring back to the lab. And you can see that here, if those two things are equal to each other, you take the natural log. Hey, the natural log of 1 is 0. Presto, affinity will be 0 when the system's completely at equilibrium. And what we're looking for are places where the affinity is positive, because that tells us that there's there's energy that could be, that is available for that reaction to proceed from left to right as we're writing it. So just to show you a few examples here from Peter Canovis's work, here are affinities. Now, the units here are kilocalories per mole of electrons involved in the reaction. And we do this so that we can compare everything on a similar basis, since there's different numbers of electrons used in the reaction. So here's energy available from hydrogen oxidation across this, these samples from Oman, and you can see that these you wouldn't know this unless you've been doing these sorts of calculations, but these are very large positive values of affinity. So ample energy availability for hydrogen oxidation. Another example is methane oxidation, aerobic methanotrophy, abundant energy available for that reaction. You can see sort of a trend of increasing energy as you go up into higher pH, where the fluids are dominated by the serpentinizing fluids. That's even more dramatic when you look at carbon monoxide oxidation. So all these reduced constituents, the hydrogen and carbon monoxide, methane coming up with the serpentinized fluid, all represent wonderful sources of energy when they can be combined with something like oxygen. Energy, of course, coming from a reaction and not from an individual constituent. Much lower amounts of energy are available for things like methanogenesis. 0 here, look at that. Hey, there's equilibrium. And there's the distribution of points, which is slightly above that equilibrium value, which means there's a little bit of energy available. And the question you might be asking is, well, is that the best that the system can do to support methanogens? Or is this everything that's left after the methanogens have done what they can with the system? And what we're finding is that there are methanogens present in the sediments associated with a lot of these springs. So we're thinking that this might be what's left after they've worked over the system. Another example like that is the anaerobic methanotrophy here with balancing the reaction with sulfate. Again, a very small and positive amount of energy, and the question remains whether this is just the remnant energy that's left after biology has worked the system. So if we go out to a pH of 11, just as an example, to get something out close to the serpentinized fluid, we can get this sort of thing. This is a ranked diagram of affinities with the carbon monoxide oxidation, at least in the Oman system, being a nice big source of energy, followed by these various other oxidation reactions on down through reactions that we consider based on geochemistry, and on down into the really sad, pathetic leftovers of sulfate reduction, methanogenesis, and anaerobic methanotrophy with sulfate. So we can generate these sorts of images of the supply of energy based this way by comparing things on an electron basis, or going to individual samples and evaluating the energy supply in each one. And what we see is a wide spread and what energy availability, and what remains to be seen is exactly what all is controlling that. These reactions are predominantly, this list of reactions is predominantly things that we know that there are microorganisms in serpentinizing systems that do these. We've added a few reactions that we think are likely based on what we've, based on the geochemistry of the system, like the Ammonox reaction, the Anamox reaction here, ammonia oxidation, the water gas shift reaction and so forth that we think are plausible, and that those metabolisms could be explored, I think, with some, it'd be worth doing. So what I've tried to do real quickly here is show you how you can go from collecting analytical data, geochemical data on these systems, doing some calculations, and having now a menu. This is the menu. This is what's on the menu here for these reactions in terms of their relative energy supply, more positive being more energy. And that gets us one step closer to trying to having an image of how geochemistry of the system is supporting the microbiology that's present. And I'll leave you with that.
Hey, thank you, Everett. So we've got some specific questions in the chat window that we might have time for, and I'd like to start though with the general question, which is to ask you if I can summarize your final conclusion there accurately, which is that because you're measuring things at the surface that are mixed with the surface, if your energetic calculations show a very small amount of energy, that might actually be an indication of something that is most likely to actually be occurring in the subsurface. When you have additional information, of course, that you can use to interpret that information. Is that totally off track?
I think that's likely to be the case, and I think these things that we think of as something like methanogenesis and autotrophic methanogenesis, where we think of that as I think of that as sort of a reductive process. I guess I'm obsessed with the carbon. That I think that's much more likely in the subsurface as long as the CO2 can infiltrate into the subsurface and it's brought or is present as carbonate minerals. So there could be a complex history, which I don't doubt, in these systems, where at one point you have fluids infiltrating, bringing dissolved carbonate into the system that precipitates as carbonate minerals, and then things change later, and there's a supply of, say, hydrogen into where those carbonate minerals are, and suddenly those carbonates look pretty much like pretty delicious with all that hydrogen around. And so I think there are probably microbes, using solid carbonate as their carbon source, and I think people are having success showing that sort of thing.
Okay, thanks. And then before we move on to the break, let's get to at least one of these questions. Darcy asks, have you looked at the transformation of iron, or are those reactions, do they suck so much they're not worth mentioning on the plot?
It's well, we're in the pH range where iron oxidation is definitely Fe2 to Fe3 in solution is so fast abiotically that it's definitely an example of something that is bursting into flame and is not good to eat. It is just simply, we can't measure the reduced iron in the water in the field. It's below our detection limit, and the rate would be so outrageous for the abiotic oxidation in these systems that I don't think there's the oxidation of iron is something that is supporting life, at least in the solution. Now that could be very different in the minerals because you're going to have the kinetic inhibitions for the reactions and allow the ferrous iron to stick around. But we have not included those in this particular analysis, but we certainly can for writing reactions for the various ferrous iron minerals and the ferric iron mineral products. But they're not as dramatic as the rest of the system.
And then Alexis asks, what is the source or process for the ammonium?
Sure. I don't know. It increases much higher in the serpentinized fluid than it is in the surface water. So I don't know why that is in terms of like the nitrogen cycle, but it certainly seems that nitrogen going into the system is coming back out as ammonia, but it's also enhanced in its concentration relative to the surface water by quite a bit.
Cool, thanks. I think the discussion will continue in the chat window, and now I think maybe we should have our coffee break.
All right. Well, wonderful. Well, thank you, Billy, and everyone for organizing this. This is a really interesting session so far. We've covered some interesting material going from the seafloor to Oman. What I am trying to cover here is telling you a bit about what we found out about the microbiology of serpentinizing systems, particularly in continental environments. And so this is some stories from our travels to various serpentinite settings throughout the world. Now, am I moving the slide okay there in terms of what you're seeing?
Yeah, Matt, I recommend you you click the down arrow
I
to
see.
the lower
Okay.
left of your slides, and then it'll page more like a slide.
Got it. All right. Now I'm clicking through everything all at once. Wonderful.
There you
All
go.
right. So you've already heard something of the Lost City, and it's really been the hallmark site for serpentinization over the last decade or more. In terms of the exciting, contrast to high temperature black smoker types of hydrothermal systems, the high pH fluids, relatively high temperatures, copious amounts of methane and hydrogen, and associated potentially abiogenic organic molecules. So there's been a ton of study at the Lost City. We've really advanced our understanding of the microbiology of seafloor serpentinizing systems anyway through the study of that site. But the fact of the matter is, beyond the Lost City, there haven't really been that many studies of seafloor serpentinizing systems. In recent years, there have been, analog systems discovered in New Caledonia region and in the Western Pacific. But by and large, this is relatively a new area of study, and there's relatively few of these systems that have been explored. And so, given this, gap in our knowledge over the years and of serpentinizing sites, and the fact that eventually much of the seafloor material ends up on land through plate tectonics, we and others went about studying continental serpentinizing systems, so Ophiolite complexes. So one of the most beautiful examples of this, in my opinion, is the Tablelands Ophiolite. This is a picture of the Tablelands in Newfoundland, Canada. I believe Billy mentioned this briefly earlier. So this is nearly a 500 million-year-old serpentinizing ophiolite, which is still seeping high pH gas-rich fluids and is a continental analog to some of what we've seen in seafloor serpentinizing systems. So as I mentioned, following the work at the Lost City, before more cruises went out there, we and others, including many of the people on this telecon, have studied serpentinizing sites in Ophiolite's continental settings throughout the globe. And so places like, in California, in Newfoundland, in Liguria, in Italy, in Oman, and so on. So, racked up a lot of frequent flyer miles and spent a lot of money on field gear for sampling and analysis of these various sites. I recently came across a paper from 2007 by Oz and others about serpentinizing ophiolites, and that map is shown in the lower left here. They were looking at geochemistry of serpentine soils, but this just shows, in addition to the dots on our Google Earth map, how many hundreds of other serpentinizing settings there are throughout the globe, particularly in continental margin type of environments. All right. So what I think is exciting about this, in addition to the fact that there are so many sites to investigate, is that these continental serpentinites cover a broad range of different conditions. It's sure they have some things in common, like the high pH and the presence of the gases and the generally reducing conditions, but they cover a great spectrum of conditions in terms of climate, in terms of hydrology, groundwater, recharge and discharge and residence times, things like the geochemistry of these systems, the geologic setting, some of which was alluded to on that earlier slide about when exactly these ophiolites arrived on the continental margins, and then consequently microbiological processes. So in the context of astrobiology, I think this provides us with a tremendous opportunity to look at some different settings, some different occurrences, which may be analogs to things we may observe on Mars or elsewhere in the solar system. So I just wanted to point out a few of these examples from our travels to various serpentinites in the course of this talk. Further, they're also significantly easier to access and cheaper to access than seafloor systems. And so some of what we've been able to do in these continental serpentinites is to go about and conduct time series studies or to develop new methods to apply to some different sorts of systems. All right, so one of the major sites for continental serpentinization that's been studied over the past several years has been the Cedars in Sonoma County, California. This is a place where Ken Nielsen's lab, as well as Shino Suzuki and Penny Morrill, have all done some work. So this is a very extreme environment characterized by copious amounts of gases, particularly hydrogen and methane, very reducing conditions, high pH groundwater, and extensive travertine deposits shown in the left-hand side of this slide. So really impressive carbonate deposits associated with circulation of serpentinizing fluids through the system. And so Shino and Ken and others have explored the microbiology of this system and found that the communities that inhabit relatively shallow portions of the system, perhaps more influenced by mixing and shallow groundwater, are of different communities than those in deeper groundwater. So they see organisms like beta proteobacteria commonly in the shallower parts of the system at high pH, and organisms like Chloroflexi and candidate division OD1 in deeper groundwater, perhaps more representative of end-member serpentinizing fluids. My group and others, including people like Don Cardis, Tori Holler, and Tom McCullum, has done a lot of work at what's known as the Coast Range Ophiolite Microbial Observatory. This is located a relatively short distance away from the Cedars in reality, but it's near Lower Lake, California at the McLaughlin Natural Reserve. And what's been great about this site, in addition to it being at a field station where we have access to a lab and facilities, is that we were able to recover drill core from this location in 2011 with support from the NAI, and subsequently establish 8 new monitoring wells in the system to conduct comparative experiments, or conduct experiments to compare microbiology and geochemistry and hydrology within the system, and to learn something more about the native populations in the serpentinizing groundwater. So some of this work was published, as far as the operations, was published in 2013 by Don Cardis in Scientific Drilling. So here's an example of Mike Kubo and Katrina Twing and a couple others putting experiments down into one of the wells at Chromo to look at colonization of different minerals within the groundwater as a function of time. But just shows an example of the fact that you can go out and manipulate these systems and do experimental work. So there are challenges to these systems, as you may have already gained through the last couple of talks. So we have high pH, low EH, so very oxidant-poor environments in many cases, and the possibility for nutrient limitation. So things like nitrogen and phosphorus, perhaps, but also the availability of dissolved inorganic carbon and the possibility that different carbon sources are necessary for organisms to be sustained in these environments. One of the things that has been seen in both of the systems I described, as well as elsewhere, is the ubiquity of certain types of microorganisms in these environments. So Shino Suzuki published a really nice paper isolating a new type of organism called Serpentinimonas from the Cedars. She published this back in 2014, but very similar strains or similar sequences related to Serpentinimonas have been observed in high pH environments around the world, places like the tablelands of Canada, like deep groundwaters influenced by serpentinization in Finland at Outokumpu, even in places like contaminated steel slag site, industrial waste location in the Midwest, work that Wes Swingley and others are doing. So this is a very ubiquitous species of organism in the beta proteobacteria that's been found at all of these sites. And what's really interesting about this is not only its ability to stand some of these extreme conditions, but its ability to utilize diverse carbon sources. So one of the fascinating aspects of this organism is that it's actually been shown to use solid calcium carbonate as a source of carbon in growth experiments. Others, including my laboratory and Billy Brazelton and several other groups, have taken metagenomics approaches to serpentinite-hosted communities. So, in other words, what this means is going about extracting the genomic DNA from the natural communities in these systems and studying the genetic content of their genomes to try to infer aspects of their function and their functional potential. So some of this first work we did in this regard was at the Tablelands, published in 2012 by Brazelton, identifying genes involved in hydrogen oxidation and carbon monoxide utilization from the metagenome in that context. And then recently we've looked at a series of metagenomes that we have from the chromo site and various types of catabolic or genes related to various catabolic reactions, including hydrogen oxidation, carbon monoxide oxidation assimilation, and sulfur metabolisms, and map those out versus pH at the chromo wells. And so that's what you see in the lower right hand of this figure is the diversity of genes and functions in some of the wells at very high pH. So supporting this idea that hydrogen and carbon monoxide are important, and even potentially acetogenesis, which is why I was asking Everett about this in the window just a few moments ago. All right, but so again, these are just a few examples of ophiolytes that are out there. Everett's mentioned the Samael Ophiolite in Oman, this world's largest ophiolite exposure, where you have exposed sections of oceanic crust and upper mantle. And there's an ongoing drilling project in Oman, including many members of the NAI-RPL team, like Alexis's group, to take part and participate in this project and study both the organisms in the groundwater as well as the biogeochemistry of core samples coming up from very deep within the system. There's also a really interesting site that's been studied lately in the Santa Elena Ophiolite in Costa Rica, which is a tropical serpentinizing environment characterized by very extreme seasonal wet-dry cycles, so short residence times, rapid recharge. And one of the things that we've observed through sequence-based studies of this system are that there are relatively abundant methanogens in that environment. And so perhaps there's a link between these short recharge events and the possibility for biological methanogenesis in these systems. So it's in the context of what we've talked about over the past couple of presentations, that's a really interesting variation on this theme. So, you know, in terms of astrobiology, what are some of the interesting things that have come out of this work? Well, we've learned something about adaptations to high pH environments, to serpentinizing environments. High pH in itself does not appear to be an impediment to life in these systems, but there is a relatively few taxa we see, relatively low diversity that are common in global serpentinizing systems, at least from the reports that we're aware of. It also seems that growth of some of these organisms is stimulated by fluid flow and mixing, that perhaps this brings in the nutrients or the chemical disequilibria that's necessary to really cause sustained growth and blooms of organisms in these serpentinizing systems. Second point is that methanogenic archaea are surprisingly absent in many of these continental serpentinites. Does this mean that there's a pH limitation? competition with the cetogens or other factors limiting their growth. I think that's still a work in progress until we get more information about the physiology of these organisms. But it's really been hit or miss in terms of seeing archaea in many of these types of systems. And then what sort of strategies do organisms have to gain nutrients and oxidants, particularly carbon species in some of these systems? There's some evidence that organisms are fermenting organic acids in some of these environments. And again, are those organic acids derived from sedimentary input, from preformed organic matter, or from abiogenetic organic matter? These are all questions that are still out there. A second interesting thing that's come about is for many years people have talked about serpentinites and serpentinizing systems as places that were likely quite common geologically on the early Earth, and that perhaps sustained some of the chemical reactions important in prebiotic chemistry. Places like the Lost City type of system where you have thermal and chemical gradients and compartments facilitating the origins and early evolution of life. At the same time, there was a paper recently, this past summer, that looked at the phylogenetic analyses of published genomes of microorganisms, not just extremophiles, not just serpentinite-derived organisms, but published genomes in general to try to boil down what some of the common physiological functions were of these organisms. And a lot of the genes that came out of this analysis by Bill Martin's group showed that many of the genes that were observed were also very parallel to sorts of things we're seeing in modern-day serpentinizing systems. So I think from both the bottom-up and top-down sorts of approaches, it's kind of converging upon these serpentinizing environments. And then thirdly, just pointing out that biosignature detection is both very challenging in these systems, but also could be very informative, and particularly in relationship to how we might conduct the same sort of analyses on other planets and moons. So generally, you have very low cell numbers in a lot of serpentinites. Places like the Cedars are very, very low cell numbers in them, sometimes in the range of 10 or 100 cells per milliliter. You have overlapping microbial, thermogenic, and abiogenic sources of some of the compounds, including methane. So to figure out how to decipher those various sources, how to tell different sources apart from one another, and what mechanisms are being used. Here, on the left here is some work recently out of Shuhei Ono's lab at MIT. This is David Wang's paper, and showing some of the different methane isotope log values that were obtained from methane samples at both the Chromo site and the Cedars, showing quite different values depending upon. Here, let me figure out where the arrow is here. Okay, so the Cedars is right here. Oh, I'm moving this around. Maybe that's not worthwhile. Well, in any case, you're
Matt,
seeing.
Sorry to interrupt. If you want to just click on the slide where you want the arrow to go, it'll behave a little nicer.
I see. Okay. Well, in any case, the methane isotope log values of the Cedars, which is where my arrow currently is, versus Chromo, and I switched slides. In any case, are quite different, suggesting different contributions of either microbial, thermogenic, or abiogenic sources are of methane. And thank you, Shuhei, for piping in there with some information about which is which. I appreciate it. So various sources of methane contributing to the isotope log signatures in these settings. Also, there's a potential to look at some different dissolved organic compounds or metabolites in these systems, perhaps things like organosulfur compounds or other organic acids would be worthwhile to investigate in terms of biosignature detection. And continental serpentinites provide another environment that we could look at these sorts of effects. And then finally, I would say that consideration of seasonal or episodic effects are also important. So to understand issues related to fluid recharge, fluid efflux from these continental springs, and how those might parallel things we observe on places like Mars would also be an interesting area of study, the temporal aspect. And then finally, I just wanted to point out that serpentinizing ophilites are not the only sites of ongoing serpentinization, that there are actually ultramafic intrusions in various settings that may provide an interesting comparison to some of these paleo seafloor that's being serpentinized. So places like the Cabeça de Vide, serpentinite influenced wells in Portugal that Igor Tiago in Portugal is looking at have, in some cases, very similar characteristics microbiologically to what we observe in Ophilites. So abundant Clostridia and beta proteobacteria. In other cases, these can be very stagnant sorts of environments, so less influence of surface groundwater and recharge. Another site that's been looked at recently in collaboration between my lab and Barb Sherwood-Lawler up in Toronto is looking at serpentinizing kimberlites up in Kirkland Lake in Ontario, and where you have these deep ultramafic rocks coming near the surface completely in a different geographic setting from the Ophilites, with some interesting parallels and some interesting contrasts to what's been observed. So I think I'll leave it at that, and hopefully have a chance to answer some questions related to this, and I hope it's given you a flavor of what we've seen in some of these systems and leads well into what Beth will be talking about.
Great, thanks, Matt. I think Ava asked a good question that would be good to start with, which is that alkaline lakes are some of the most productive ecosystems on Earth. Why are serpentinizing settings so much less productive? And perhaps as part of your answer, you could give us a brief explanation for why high pH is a challenge to biology.
Yeah, I mean, I think this is still a work in progress, but high pH is a challenge to biology, at least from what we know in the literature, because many organisms require proton motive force to generate energy, to generate ATP. And so the fact that you have protons being extremely unstable in the extracellular environment is probably a first-order challenge to this whole situation. And so they need to go to great efforts to generate some sort of force, and at least in alkaline lakes in many cases, there is the potential to use other ions as a source of generating this gradient, whereas in many of these serpentinizing systems, you're dealing with very freshwater conditions that are not providing those ions for generation of those gradients. But I also think, just thinking back to what I think it was Everett was talking about, is perhaps some of the chemistry we're seeing is not so much because of some active metabolism, but it's because of what's not being consumed. Perhaps the lack of productivity in some of these systems is leaving compounds like ammonium behind. And so we also need to consider that in terms of the context of the biogeochemistry we're seeing.
Yeah, thanks for the answer. Earlier, Beth Orcutt asked whether you could elaborate on the mechanisms that are being used to access carbon from solid substrates.
I don't know much more about it than what was published in Shino's paper, that they provided some sort of fine-grained suspension of calcium carbonate that these organisms were able to utilize, and it seemed like they were attached to the particles, from what I recall, so that there's some sort of interaction between the solid surfaces, the organisms and the solid surfaces. But in terms of the fine-scale mechanisms, I'm not entirely sure. Although Annie typed in, it looks like, and perhaps she has more insight into that issue.
Thanks. And John asks whether we have a sense as to what the biosignature preservation potential of the carbonates are. Do they preserve organics or biogenic textures or structures?
I mean, I think even dating back to what we saw in the Lost City chimneys many years ago, it certainly looks like the the filaments and the biofilms serve as a template for precipitation of some of these carbonate phases. So that in itself could be an interesting avenue to explore in terms of biosignatures and serpentinizing systems. In terms of the ability to preserve organics or biogenic, I guess, organics is the main thing. I don't know for sure whether serpentinizing systems are more or less prone to preserving organics than other types of systems. I would think that organisms in these systems, living organisms, because they're so desperate for carbon, might have strategies to extract any edible carbon out of some of these organic structures that are available, but that's pure speculation.
Thanks. And thank you, Annie, for elaborating on these issues in the chat window. That's what we want to use the chat window for, so cool. Maybe that's a good time to move on to our fourth invited talk. Thank you very much, Matt.
All right, thank you.
So fourth invited talk of the day is from Beth Orcutt about drilling into serpentinites. Go ahead, Beth.
Okay, Beth, you're the presenter now, so if you could start your webcam and take it away whenever you're ready.
Beth, it looks like you're still muted.
Beth, do you need the instructions to call? I'm going to pull those up for you real quick. So just follow the top set of instructions. So mute Adobe Connect and then call the number you'll see on your screen. Okay. In the meantime, Billy, are there any other questions we didn't get to? Maybe we could address?
No specific questions yet. There's still an ongoing discussion in the chat window, which is great. Maybe now is a good time to put out another call for pop-up talks. We're full for today, but I believe we have openings for pop-up talks tomorrow and the third day. So please send those slides to Mike if you'd like to give a 1, 2, or 3 slide presentation.
Hey everybody, can you hear me?
Yay.
Sorry, I tried to unmute my phone and I hung up.
Okay, Beth, your slides are coming back up. Take it away whenever you're ready.
Okay, great. Well, thank you to the organizers of this workshop for inviting me. It's been really fun to listen in on these talks. And I'm excited to talk with you all about some of the recent work we're involved in trying to explore the serpentinization that may or may not be occurring in the subsurface in the area of Lost City. So for those of you who don't know me, I'm Beth Orcutt, a scientist at the Bigelow Laboratory. And the work that I'll be talking about is related to an ocean drilling expedition. So IODP is the logo up there. But there's also several partners involved in this work, including our NASA NAI Life Underground project, funding from CW and the Deep Carbon Observatory. And so I want to talk to you today about some of the challenges and fun things that we're learning from this system. So let me see how this works. Okay. So this is probably no surprise to everybody participating in this workshop, but I'd just like to put it here to remind myself why we're so excited about this and thinking about energy sources for life that we could be looking for in other environments. That's kind of what got me excited about this field way back when I was a student, and I'm still excited about it now. And as several others have already shown, and just to reiterate here to the team, we're looking at how the fluid rock reactions occurring with these minerals in the mantle and mantle rocks can lead to the formation of chemistry and conditions that could support life independent of photosynthesis. But also some of those, the chemistry of some of those reactions also leads to challenges for life. So we're trying to tease that apart. And we really want to get into the rocks where this reactions may be occurring and not just looking at the surface expression of those processes. And so as Billy has talked about, and Matt and Susan, the Lost City vents, the beautiful vents there where you have these carbonate chimneys, are giving us a mixed signal of what's happening in the subsurface and what's happening influenced by the seawater that's around it. But the motivation for the project that I'll be talking about is to try to get into the rocks where these reactions may be occurring in the modern day, to see what microbiology may look like in this environment and how microbiology may affect the reactions that are occurring. So just to reorient you, in case you are not familiar with where the Atlantis Massif is, or where Bigelow is, which is where I'm located, we're working on the western side of the Mid-Atlantic Ridge. And to zoom in on that environment and talk a little bit more about how it was formed, I'm not sure where this black square came from up here in the top left, but that's okay. To orient you, in this bottom image, north is pointing towards the bottom right-hand corner. And the idea here is that as the plates are pulling apart, tectonic processes have led to the uplift of mantle rock in this mountain that forms the Atlantis Massif. And there had been drilling previously in the central dome of the Atlantis Massif, mainly recovering gabbros. And the motivation for this recent drilling expedition was to really go into, this southern end of the rift. The Lost City vents are basically just on the back side of this, to get into these rocks and to drill a transect across this environment where rocks on this end were potentially deeper in the crust and haven't been exposed for the shortest length of time at the seafloor, as opposed to rocks over here, which were shallower and have been exposed for longer periods of time. And to go across this gradient and collect rocks to see what processes may be occurring related to serpentinization. And so we went out and drilled this expedition in October 2015 through December 2015. And typically with a drilling expedition, you would go out with a drill ship and lower a giant drill pipe into the seafloor and core into your environment. But that was not going to work for the Atlantis Massif for multiple reasons. One, it's relatively challenging to drill right into rock at the seafloor, which we knew the environment would be like based on submersible dives in this environment. But also there was a strong desire to recover these shallow sequences at the very top of the Atlantis Massif where these processes were going to occur. And with traditional drilling, like done with an ocean drilling vessel, you would usually get 10 to 20% recovery if you're lucky. And often the rocks are very broken up and fractured. And so we wanted to use a drilling platform that would allow us to get very high recovery in these upper carbonate cap sequences. And so the way that we did that on the Atlantis Massif expedition was to use what's called a seabed drill. And so the idea here is that you have a self-contained drilling system. Here's a cartoon of a person for scale. And this system would be lowered down from a research vessel to the seafloor, connected by an umbilical that runs power, video, etc. All the drilling is done in place. All the cores are collected into a magazine. And then the entire drill rig is brought back onto the ship. And just to show you what this kind of looks like, it's all controlled from a little van, kind of like driving a video game. And you can also see what the seafloor looks like, which is actually very helpful in our case, because in some cases we could see bubbles coming out of the seafloor as our operations were occurring. So there are several sea drills that exist that are used in industry, and there's also some that are more specific for scientific research. And so the two that we used, one is called the MEBO drill. It's from Germany, from Bremen and the Marum team. And then there's another rock drill that we used called the Rock Drill 2 that's operated by the British Geological Survey. And again, the motivation for this was to use these systems so that we could get higher core recovery than would traditionally be possible with conventional drilling. We also wanted to soup up these systems to do more than they had ever done before so that we could really understand if serpentinization was occurring and generating methane and hydrogen and changes in redox chemistry. And so as part of this expedition, there were several technology developments that our team was involved in to make these drills even more capable than they already were in collecting cores. And so one of those was that we wanted to add the capacity for collecting bottom water as well as water that would be coming out of the borehole as we're drilling so that we could see changes in fluid chemistry. We added on several in situ sensors that would also be sampling this environment as we're drilling and allow us to sense things like methane, redox conditions, pH, and also dissolved oxygen, conductivity, etc. So we designed a system that could enable us to do that. We also designed a system that we could mount onto the drills where, unlike a traditional drilling vessel, where as you're drilling, you're pumping down muds and things to help with the drilling process, since all of the drilling is done on the seafloor, self-contained, we needed to come up with a system for delivering synthetic tracers as we were drilling so that we could assess the quality of the core material we brought back for doing microbiological analyses. So we came up with a small pump system to inject a synthetic tracer during drilling and showed that that worked quite well. And then finally, one of the things that we wanted to do was not just drill a hole and collect core material and then leave. We wanted to be able to seal off the holes that we drilled with instrumentation that would allow us to come back with an ROV or a submersible and directly sample the fluids in these boreholes, because that fluid would give us the best indication of what the fluid chemistry is like in these environments. Whereas the water coming out during drilling is highly mixed with seawater during the drilling process. So we also came up with a system for these borehole plugs. So I'm going to show you a little bit of some of the data that came from some of these technology developments, but there's more information available in the preliminary report about this expedition if you are interested. Just to kind of show you what this looks like instead of just a cartoon version, this is an image taken from some of the cameras on the rock drill, one of the rock drills while we were drilling. And it's showing here kind of in a snapshot, but if you can imagine, this is a video of fluid coming out of the open borehole as we're changing around drill pipe, showing that there was advection coming out of the system. And so in boreholes like these is where we would install these borehole plugs and aim to go back in 2018 to collect samples and do some additional work. So again, here are some of the sites that we drilled. They have these wonderful site names based on the Ocean Drilling Program naming system. And one of the exciting things for us is that the team of scientists involved in doing both the fluid chemistry as well as collecting samples of rocks from drilling were able to see that in some of the sites, there were elevated concentrations of methane and also hydrogen in the waters that were collected during the drilling process, and also in some of the rock samples themselves. So this indicates that there are hotspots of methane and hydrogen leaking out of the Atlantis Massif and giving us an indication of where hotspots of serpentinization processes may be. This is relatively new data. There's a lot more work to be done looking at isotopes and things like that to figure out where this methane and hydrogen may be coming from. But I just wanted to give you a little taste of what we've found so far. So some of the other outcomes, and I'm sorry, it looks like the picture I had of Yuki Morono doing cell counts didn't show up. But some of our outcomes from the expedition so far, and again, this is less than a year old, is that we got really excellent recovery, although we didn't get as deep as we would like, of these carbonate capped sequences on the top of the Atlantis Massif. And so the petrologists that are part of our team are very excited to tease this all apart and learn about the processes of serpentinization and fluid rock reactions and alteration. So stay tuned for that. We were able to demonstrate on this expedition that these new technologies for assessing the quality of our samples seem to work really well. And I think these are important topics to consider for designing systems to go to other planets and how we assess the quality of those materials that are brought back. Because of doing all this work to show that we can limit contamination, we also can show that we really collected some great samples for doing microbiological analyses, and several of the scientists participating in this workshop are involved in doing some of those experiments to look at which microbial processes are occurring and how they might be affecting fluid rock reactions. And as I mentioned before, we also see this evidence of methane and hydrogen leakage across the Massif, and that needs to be connected with these more detailed analyses of the rock cores to try to figure out if the serpentinization processes are occurring in the modern environment or if there are indications of past activity. And so as I've alluded to, there are many, many, many scientists involved in this expedition. All of this work is ongoing. We're actually still in the moratorium phase for this expedition, but the expedition reports will be coming out within the next few weeks. So there'll be lots of material about all of the rocks that we collected in the offshore phase and then started examining in the onshore phase. So I just wanted to say thank you to all the scientists that were involved. And just to wrap up, kind of the big motivations for this expedition is kind of taking what Matt and Billy and Susan and Everett have talked about. You know, what is the life like on these rocks? Is it relatively unique? Is it going to look like what we find in tablelands and chromo and other continental systems, or is it going to look more like Lost City, or is it going to look like something totally different? How does that life there and where it is indicate what's going on in the rocks themselves? So there's a lot more work to go on here, and I'd encourage you to stay tuned. And this is our expedition website where information will be presented as it's available. So I try to keep it short and sweet because I want to see some of these pop-up talks and save time for them. So I'm happy to take your questions.
Thank you very much, Beth. We have one question so far. Are there any plans for a long-term observatory at the Atlantis Massif?
So thanks for that question. During the expedition, we were able to successfully install 2 of these borehole observatories in 2 of the holes. There may be 2 other ones that are installed. We're not sure of the quality of those installations because it was really like you pull off the drill rig as you're installing it and kind of leave it blind. So we won't know until 2018 when we go back how well they worked. But the idea is that this infrastructure is there and in place and could be used as an observatory should there be interest in doing it and should the infrastructure work the way that we designed it. And if you have more questions about that, Susan and Billy would be good people to ask because they're part of the expedition to go back there.
Yeah, stay tuned. Don Cardes asks if there's any chance of using the methane probe technology at other sites.
Yeah, thanks, Don, for that question. So the methane sensor that we use is actually a commercial product available from Granitech. It was designed for use in other systems, oil field type work. And we basically incorporated that methane probe into a Seabed CTD package to measure the concentrations. And I should say that Marv really led a lot of that work. That's his field of expertise. So that sensor exists. People can buy it. The one that we use is currently at the British Geological Survey and with the MEBO team, and it could be part of the kit for those rock drills should people want to use those rock drills for doing additional work.
Thanks. I have one more question, and while I ask it, I can say that people are welcome to ask questions for any of the talks today before we move on to the pop-up talks pretty soon. So my question is continuing with my proposed theme of how to make measurements at the surface in order to understand what's going on in the subsurface. Would you say the rocks that we recovered with this expedition are more or less representative of the subsurface than the Lost City chimneys, for example?
Yeah, that's a fun question to think about. So, and for people that are interested in this, I think the expedition reports will help tease this apart. Some of the sites that we drilled, if I can go back here in my slides, one more. So some of the sites that we drilled, we can see from the assemblage of rocks that we probably are not, we didn't core into an in situ sequence, but are actually looking at more of physical movement of rocks and collection of them in a place. And then in some other sites, we do have a nice sequence that looks like we're looking at an in situ set of rocks that haven't been moved around to somewhere else. And by looking at the alteration chemistry of those rocks, Lisa and others on the phone may be able to give even more indication of that. We can give an indication of probably both this downwelling process, seawater coming into the system and reacting and precipitating carbonate, things like Susan talked about, but also deeper processes are occurring. So I think even in the Atlantis Massif, where our deepest site is 16 meters, we're still looking at mixing processes between seawater infiltration into these systems, as well as processes that are coming up from below. So it's Lost City, but a little bit deeper, in my opinion.
Yeah, yeah. I think it's a tough question in all of our minds. Certainly, the bubbles and the flow coming up are really encouraging. Any more questions? If not, we can move on to the pop-up talks. Are those ready, Mike?
Yes, they are. First up is Katie Rumford. Just give me one moment.
Okay. Thank you, Beth.
No problem. Thank you.
All right, take it away.
Okay, so today I'm going to be presenting some exciting new results from the RPL NEI's team work in the Samael Ophilite in Oman. And as we've been discussing all morning, serpentinizing systems can sustain microbial life through the abiotic production of hydrogen and reduced carbon compounds. However, one of the major questions remaining is, are there actually organisms subsisting off serpentinization reactions in the subsurface of these systems? And in Oman, we have a unique opportunity to study this because we can sample deep fluids from a series of wells drilled by the Omani government to search for water resources. And these wells are up to 300 meters deep and span the crust-mantle transition, so we can sample from various lithologies such as peridotite and gabbro, and also various hydrological regimes such as shallow and deep flow. So this allows us to address even another question: if there is life, how is life distributed in the subsurface of a serpentinite? And answering this question will be an important first step towards developing any predictive models for biological habitability in these water rock reaction systems. Next slide. So we collected 20 deep fluid samples over 3 years, and on the left here is a principal component analysis of these geochemical parameters. And you can see that we observe highly variable fluid chemistries throughout the Ophiolite, and we find 4 main fluid types. We see a fluid type that is very hyperalkaline, calcium and methane rich, hosted in peridotites. We see a second peridotite fluid that is more alkaline in pH and higher in magnesium. We see high silica gabbro hosted fluids, as well as more of a hybrid type fluid found at the contact between the crust and mantle boundary. And we think that these different chemistries are really reflecting differences in both the host rock lithology and the extent of water rock reaction. We are able to detect life through high throughput 16S gene sequencing in all of these fluid types sampled. And interestingly, these fluid types host distinct microbial communities, as highlighted by this non-metric multidimensional scaling analysis shown on the right. And so wells that plot closely together have more similar microbial community compositions. And the size of the marker reflects the richness or the number of species that we observed. And what we see here is that in the really hyperalkaline fluids, we see a distinct community of low richness. And in the alkaline peridotite and gabbro fluids, we see rich communities of similar composition, with contact fluids having its own unique microbial composition. Next slide. And so what we really have found is 4 unique habitats within the Samael ophiolite illustrated here, where there's shallow flow, whether it be in alkaline peridotite fluids or gabbro. We see a lower pH, higher DIC and sulfate and nitrate concentrations. And here we see organisms associated with nitrogen cycling, such as Nitrospira and Nitrososphoraceae, as well as candidate phyla OP3 and Parvarchiota. If fluids percolate deeper and have more extensive water-rock reaction, we see much lower DIC concentrations, up to 2.9 millimolar hydrogen concentrations and up to 2.3 millimolar methane concentrations. And here the communities are dominated by OP1, Meothermus, the sulfate-reducing family, Thermodisulfovibrio, and we also see methanobacteria. And the contact fluids likely represent zones of mixing, and like in other serpentinizing systems where there's mixing, we see a lot of Beta-Proteobacteria phyla. We think these represent areas of more chemical disequilibria. We do detect some methane in these fluids, but higher concentrations of sulfate and nitrate than in the hyperalkaline fluids. So overall, the consistent parsing of these microbial communities as a function of fluid type likely represent differential energy availability and aqueous geochemistry, such as pH, that result from these fundamental differences in rock lithology, as well as in the extent of water-rock reaction.
Thank you, Katie. I have a question about the different kinds of fluids. Were those definitions of different kinds of fluids hard cutoffs just based on your chemistry measurements, or were they more of a holistic interpretation based on your insights into the system?
So I based these fluid types based on the principal component analysis we saw with some geologic context from the wells. I mean, there is one well that we observe in this system that is on a geologic contact, but has fluid chemistry consistent with hyperalkaline peridotite fluids. And so that is a little more tricky. So there's definitely not a hard cutoff. It was just based on the principal component analysis.
Okay, thanks. Don Cardes asks, do you see free CO2 in your gabbro type wells?
So we just measured total DIC, and we see about, well, greater than 2.2 millimolar total DIC, but it's not speciated.
Right. Yeah. We have some typing going on. Don says, thank you. Jen says, can you translate this to a rough surface area or fraction of exposed ophiolite?
Insight, anyone else?
That's a tough one.
You can ask her a question.
Action exposed, well, it depends how.
So I think, well, so part of the question is, rough surface area that's totally exposed. But when it comes to peridotite, I don't think we have a good handle on how much of the fluids that are stored in the aquifer are undergoing a shallow flow versus a deep flow. And we only have a few wells that are drilled by the ministry where we can even access these fluids. And so until we get a better grip on some of the hydrogeology of the setting, it would be hard to translate at least the alkaline peridotite versus the hyperalkaline peridotite. But gabbro versus peridotite in general would be easier, but I don't have exact.
Yeah, Katie, you did a great job handling my poorly expressed question. I was just wondering to see if you could sort of rank the different fluid types based on how much exposure there might be in Samael or other ophilites, just to, I guess, prioritize where we might look for different unique environments. In other words,
so
if you
in
have,
the Samael Ophilite, I think there's 15 kilometers of the exposed sequence, and about 8 of it is gabbro. And so, but within the prototype, I'm not sure how much of it would be a hyperalkaline versus an alkaline. And so, I mean, the Samael Ophilite is one of the best exposed ophilites. So there's a lot more surface area that we're looking at here compared to other ophilites. So depending on how much of it was abducted, how much of the mantle sequence, I think it would differ site to site.
Okay, thanks.
And Jesse asks, what is the explanation for the lack of dissolved hydrogen in 3/4 of the wells?
So it's lack of detection. So it's possible that there are nanomolar concentrations of hydrogen that we weren't able to measure, and that organisms could be drawing down this level below detection. But at this point, we don't know.
Yeah, it's the
But we do detect
paradox.
hydrogen in all the hyperalkaline fluids that represent extended water rock reaction.
Mm. Yeah, I would say we always have the paradox that if you don't see hydrogen, is it because it was never there or because biology ate it? Thank you very much, Katie. We can move on to the second pop-up talk by Don Cardes.
Okay, Don, if you're on the line, if you could unmute and start your webcam, that'd be great.
Hi, this is Dawn. Can you hear me?
Yes.
Yes, loud and clear.
Oh, great. All right, I'm starting my webcam. And here I am. Hello.
You're almost there. You're probably going to have to click Start Sharing on your webcam preview.
Let's see here.
Should be right in the middle of your camera view. Perfect.
Hi. Visible
Okay,
now
and then
to everybody.
just a reminder, to advance your slides, click the down arrow icon to the bottom left of your slides. Thanks.
Okay, thanks. Thanks so much. I'm glad to be here, everybody. I know I have just a few minutes, so I just put together a short presentation here, kind of an update, serpentinization in the Philippines. And I've listed next to me Darcy Meyer-Dombard, Kristen Waichis, Shuhei Ono, and Kaloy Arcilia. So these are all members of our sort of problem-solving group here trying to understand what's happening below ground at some ophiolite sites in the Philippines. And so the backdrop here is showing you a thin section in cross-polars, showing you fractured olivine relics within a mesh of serpentine and magnetite-rich veins. And this is from our Punbato site where we visited back in 2012, 2013. So this is kind of a quick review of what we've published back 2 years ago. And then I want to give you a bit of an update. Okay, so you're in the Philippines, and there's two areas. There's many, many ophiolite bodies in the Philippines. I want you to pay attention to Zambales up in the north for a moment, and then I'll take you down to Palawan, this island that's in the lower left of your screen. So previously, we actually had described everything that's shown as a white symbol with a label. So up in Zambales, we had Manlalawag Springs, the Punbato Springs, and then a Gabbro-hosted artesian well at Big Biga. And then down in Palawan, San Isidro and Minot Falls were some very warm springs. And then since that time, we're really interested in pursuing within that sort of dashed gold circle a set of what appear to be hyperalkaline and very shallow groundwater flow systems in the Nara region of Palawan. So first, to look back, in the Zambales ophiolite, we had described for you all multiple sites of really high pH, carbon monoxide, methane, and hydrogen-bearing springs. And so the two that I think really came out as featured in our past work were the Manlalawag Spring and the Punbato Spring. So quickly on the lower left, well, the main picture is actually, you see calcite rafts right on the surface of a high pH spring at somewhere in the neighborhood of 11. That's one of our Filipino colleagues, Gama. He was a graduate student at the time. But in the lower left inset, you see two stiff diagrams just showing you for Manlalawag Springs on the top labeled ML1, the kind of sort of quick image-based major ion chemistry that we've got. So it's a calcium-rich, bicarbonate poor fluid with a sort of a salt component there and measurable chloride. Pun Bateau, which is the picture on the main body of the slide, is, you see, really dominated by the calcium as well. And they both sort of proved to be rich in serpentinization sort of influence. And now here's an additional site that we're just starting to learn about now. In fact, I'm sort of freshly back from field work down in Palawan. The Deep Carbon Observatory funded Darcy and myself to go and meet Kristen Whitecheese there to do some additional sampling. And so what we found are multiple new sites of high pH springs and some shallow groundwater regimes and some flammable gas emanating from them. So this was quite a forensic study, just kind of exploring the landscape and figuring out where it might be good for future endeavors. What you're seeing left in sort of the center of your screen, it looks sort of a bluish-green pool, is an excavation down through some chalk crusts. And the standing water is elevated in pH. It's high 10s or 11. And it has some really interesting carbonate formations that seem to be following lines of groundwater seepage down through that excavation. And let's see, I think I can, there used to be a way for me to get a pointer. I'm not sure with this new screen. Let's see here, right here. So I think you can see my green arrow. So up here would be this excavation that was.
Yeah, sorry, Don. I'll bring the slides back. Just click where you want the pointer to go, and then that'll keep your slides still.
Oh, okay, right there. So now you're seeing a green pointer into that same excavated area, and I have a handful of this organic, rich, based on color, anyway, carbonate material that was sort of crumbling right off of the edge where there was groundwater seepage. We sort of prospected around with a multi-probe meter and found that this was where there was the highest pH and the lowest ORP in the standing water. And you have all this carbonate formation. On the right, I'll see if I can click over the right. Okay, so now the green arrow has moved to the right at a new site called Governor Spring, where we had my field notebook open to my left here. We had at the source, which is shown right there, water is coming out from kind of a bedrock contact with some travertine deposits. We had pHs ranging from 11.34 up to 11.42, and some ORP data screaming down to about minus 655 millivolts in terms of raw ORP data. And so these are areas that are just begging for greater description, and some of the preliminary gas collecting that we did there will soon be characterized by our whole team here. And so I need to really recognize, so Kristen Whitecheese is with us as a postdoc in Shuhei Ono's lab, and we're really excited for all the data and interpretations we'll soon be able to provide you. So thank you. This is the closing shot, just a word on it. It's a new sort of field locality for me. That cursor can go away, although I don't know how to undo it. There we go. This is an ultramafic conglomerate, actually, and so there's some cement crusts that are sort of filling it in, and this is a real weathered spot at the edge of near to one of our higher pH steep areas. And I'm finding it really interesting that we have I tuned in after teaching just when Matt Schrank was mentioning, maybe different kinds of environments where serpentinization can be continuing. And here we have kind of landscape scale giant conglomerate with serpentinite boulders and some really interesting geochemical signals that serpentinization continues. So thank you, and I will be happy to address any questions.
Thank you, Don. If it's like someone's typing, I'll ask one question in the meantime, which is, do you know anything about salinity or conductivity of these new springs? Matt was talking about how the extremely low salinity, low level of ions in other continental systems is a big challenge for life.
Mm-hmm. Well, I have some environmental probe data that we've just collected. So the first was that sort of the chalk crust dugout that we were looking in. The conductivity was just shy of 1 millisiemens. So it was something, I'm looking at the range, it was 0.937 to 0.949 in millisiemens. And then for the Governor Spring, which was the one that I had the source pointed out and discussed, the really reducing characteristics of the source water. That one had a conductivity at something like between 1.84 and 1.89. So they're still real but low, I would say. And more on that.
Okay.
More on that when we know more.
Cool. Thanks for reporting fresh off the fieldbook results. Cool. We should move on to our third pop-up talk, but thank you very much, Don. And I believe our third one is Annie Rowe.
Great. Hello?
Yes, we can hear you. Go ahead.
Oh, great. Awesome. So hi. For those of you who don't know me, I'm Annie Rowe. I'm a postdoc with the NASA Astrobiology Institute based out of USC called Life Underground. And I'm going to tell you a little bit about some work I started as a postdoc with Ken Nielsen, looking at the cedars. So Matt gave you a nice introduction to the cedars, a terrestrial serpentinization site that we've been studying for several years in Ken's lab. And it has, very characteristic microbial community of terrestrial serpentinization systems. I'm going to be focusing on a spring that has not been well studied microbially, but it's one that we think is a mix of sort of shallow and deep groundwater communities. And so we wanted to use this site to look for evidence of mineral-reducing bacteria. But sort of as Everett mentioned, one of the challenges with working with these terrestrial serpentinization systems is we can see microbes there, but it's not always obvious what the oxidants are. So geochemically, oxygen is, we don't detect it below the surface in most of the springs. We have almost no sulfate in the system. I think we measure about 1 micromole. We don't measure things like nitrate, nitrite, or reduced iron. So one of the things we wanted to look at was the availability or the potential for microbes in the system to use solid minerals as terminal electron acceptors. But again, this is a low biomass system, and so to sort of begin to circumvent this problem, we decided to try to enrich in vivo for organisms that could do extracellular electron transfer. So we used a really simple electrochemical enrichment. So this is a 2-electrode system. It's very similar to the way a microbial fuel cell or a battery will work. We have an anode here that can serve as an acceptor for electrons from microbial processes. And then we have a cathode here at the air-water interface that can abiotically reduce oxygen. And so we incubated this at a campsite spring for about 3 months. And we could measure current over this time, so we had a data logger. And we saw that after about 20 days, we could see pretty consistent current generation. And so there may be some lag time in terms of getting the microbes to associate to the anode. And interestingly, we saw some oscillations on a daily basis. And so if we zoom in on sort of a 3-day period here, we can see that we have sort of a boom and bust in terms of current generation. And this bust, or this decline in current generation, happens about noon. So this is sort of right when we have sort of the most high sunlight. And this is sort of counterintuitive to what we would think would happen if it was some sort of temperature-driven process. So we would expect, during the day, the temperatures to be higher, so we'd actually expect more current production if this was some sort of abiotic reaction. And just sort of in the interest of time, that was most of what I was going to tell you. From here, we enriched in this electrochemical system heavily for firmicutes. And unfortunately, it wasn't easy at this point to get an isolate, but we sort of stuck with it. And we eventually got an isolate. This activity, electrochemical activity, seems to not be linked to hydrogen production, which was a little bit of a surprise. But that might have to do with the spring that we were working in. And, you know, it's functionally anaerobic because of the activity of hydrogen oxidizing aerobes, but potentially it's also pretty hydrogen limited as well by those same activities. And we had some evidence of, you know, connections with phototropes as well. And so maybe that's, there's some sort of link here. But nonetheless, we think this is an interesting approach to start looking at microbial activity. And I think in the future, we want to try this in a different spring that maybe isn't quite so influenced by the shallow community. So we have a nice isolate. We could look at electrochemical activity. And I just really quickly want to touch on sort of the energetics of iron reduction at high pH because, well, basically, if you can see, if you have an iron reduction reaction that drives all of your iron to dissolved iron, this is a very pH-dependent process. But I think one thing we have to consider as a community are these reactions that involve mineral transformations. And so these are much less dependent on pH. This is an example of maghematite going all the way to magnetite under a variety of pH conditions. And you see a really shallow slope here. Yeah, and that was all I was hoping to tell you guys about. These are my co-authors. This was done in collaboration with an electrochemist who is also a postdoc in Ken's lab when I started, Akihiro Okamoto. And yeah.
Thank you very much, Annie. Penny asks, which spring did you use?
I'm sorry, Penny. Yeah, this is Campsite Springs.
Campsite Spring. All right, thank you. I think we have just enough time to get to our fourth and final pop-up talk, but thank you very much, Annie. All right, Alberto, are you here?
Yes. Can you hear me?
We can hear you, but can't see you. So I think, as Mike says, there's a button on the center of your webcam window that you click to share.
Yeah, let me jump in.
Where
So,
is
Alberto,
it? Okay, I see.
Yeah, if you look above
Start
Billy's head, there should be a start webcam button. Great. Looks like
Okay.
you found it.
All right,
Perfect.
go
And
for
you
it.
control your slides. You just want to click the down arrow icon that's in the lower left of your slides.
Yeah, okay, perfect.
All right, take it away.
So, thank you. So if I do this, it works, right?
Actually, if you click the down arrow icon to the bottom left of your slides, that'll advance them in slides. That's better.
Okay, perfect. Okay, thank you. So the idea is to show some results about the potential role of serpentinization at high pressure conditions as a source of hydrogen and abiotic methane. So I'll start with this diagram to the left. Can you see the green arrow here? Maybe. Anyway, this is a pressure temperature diagram, and you can see in blue the stability field of serpentine minerals. That is, in other words, the stability field of serpentinite. And here to the left, there is a small circle that should extend to much higher temperature. But the idea of the circle is just to show that that most natural laboratories we have for studying serpentinization are from the very shallowest part of the serpentine stability field, and we can have almost the same processes at much greater depth, down to at least 4 GPa. And the best place to study these processes are subduction zones. So you can see to the right, the blue box that shows the region where we can have, and we know that from natural studies and from geophysics, that quite a lot of fresh mantle material is serpentinized. So the question is this deep process able to release hydrogen and produce some abiotic methane or not. So I'm going to show you some results from the Italian Alps. Here you have a map, and there are two main things to keep in mind. So you can see in the middle of the map a light green zone. This is a fresh core of peridotites, so olivine, pyroxene, and everything. And then there's a shell, a serpentinized shell, all around this peridotite body that you can see in slightly deeper green color in the map. Yeah, the two things that must be kept in mind are that the shell contains serpentinites and carbonate. And the second is that we know from several data from the literature that at least a large part of the serpentinization occurred in the subduction zone. So these serpentinites formed at high pressure conditions. I'm not going to talk about this in detail, but this is well known from the literature. So the question in our case was to see which kind of fluid was produced during the serpentinization and what was the effect of this fluid on the surrounding rock. And all I'm going to show you is from the shell. So it's from the serpentinized and carbonated serpentinite here from the shell. So next slide. So these are data that are in press, so they should appear in Nature Communications quite soon. And I'm just going to show you two details that are the two most remarkable features. So the first thing is on the picture to the left. You see maybe you cannot see the labels, but so all you can see, all the white matrix is carbonate, calcium carbonate, and inside this carbonate matrix we have some small fragments of serpentine. You can see an elongated one to the bottom of this figure, and there are many others. And all the small dots or bubbles you can see in the photo are fluid inclusions that mainly consist of methane and dihydrogen. So this is the first thing. First natural evidence, we have a lot of methane and hydrogen in these deep rocks. The second thing is on the other picture to the right. This is an SEM image showing the interface between these serpentinite fragments and the carbonate matrix. And you see that there's quite a lot of black stuff all around the serpentine class. And this is graphite. So we tried to understand how this graphite formed. And I'll be very fast on this because I don't have time, but we reconstructed based on several pieces of evidence, three main stages with two possibly new paths to generate methane at high pressure conditions. So the first is reaction one that involves antigride, calcium carbonate, and an external fluid that is dihydrogen to give some new phases like diopside, brucite, and a fluid containing methane. Then what we imagine is that part of this methane saturated the fluid in carbon and precipitated the graphite. And then there's the second stage of reaction that in our model produced some methane, that is the interaction with the external hydrogen-rich fluid and the graphite. So we believe that we have two successive reactions able to produce methane through kinds of dry reaction at high pressure conditions. So again, I cannot give you all the details, but we have several pieces of evidence for that. So here in this cartoon, you have a summary of what we believe for these rocks. So the principle is that serpentinization of the big peridotite body I showed you in the first map released quite a lot of the hydrogen. Can you hear me?
Yes, we can hear you, but we should probably wrap up soon.
Okay,
So
I have some problems on the phone. So the idea
so
is that
30 more seconds or a minute. Thanks.
Okay. The idea is that serpentinization of the big piece of fresh peridotites released a lot of hydrogen, and we found some hydrogen preserved in fluid inclusions. And then the interaction between this hydrogen and other rocks just next by the peridotite bodies produced a lot of methane. So then, just for conclusions from our study, we estimated the depth at which this methane is released, and we can say that it happens between 40 and, let's say, 15 kilometers depth during the exhumation of this body. Then the duration of this process, based on some petrological evidence, we can say that methane was produced over 0.5 to 1 million years. We don't know if this was episodic or continuous, but we have good constraints for that. And then the fluxes, based on the mineralogy and the reactions, we estimated that more than 350 kilograms of methane were produced for a cubic meter. And we also got some estimates on what could be released at subduction zones from this kind of deep serpentinization processes. That is in the order of 0.8 megatons of hydrogen annually and 1.5 megatons of methane. So these are possibly two new and alternative pathways to produce methane at high pressure conditions. It is true that this is from subduction zones, and it is not sure that the same process may take place in other planets, but the message is that from high pressure rocks we can have some alternative ideas of the reactions, the abiotic reactions that can produce this kind of gases. Thank you.
Thank you very much, Alberto. That's really interesting. You have some questions in the chat window that if you could answer them via typing, and then we can call a close to our session for today, and we can carry on the conversation in the chat window. But thank you very much. Would Alexis or Jen and Tori like to do a wrap up for the day? Or should we just call it and reconvene everybody tomorrow morning?
I will just say it was wonderful to have such excellent participation today. And again, knowing the topics and the speakers tomorrow, I'm not sure if Jen or Tori have heard from you or those organizers, but if you are interested in doing a pop-up talk, even injecting ideas, particularly in a really brief format to get them on the table, it's again welcome. We look forward to it.
Thanks, Alexis.
Yeah, I think we can just quickly wrap up from our end by thanking everybody for the great talks today. And in particular, it was really pleasing to see the pop-up talks and how well those work. So as Alexis said, please do chime in. We have still a couple of open spots, I think, for the next couple of days.
And related to that, we're going to put a link to a survey that we try to do after all of our events, because we'd love to get your feedback on what you thought about the technologies, how we integrated it, the whole production, and also topics, if there's any upcoming topics that you'd like to suggest that we cover. So please take a minute and fill in the survey. If you know people that have already dropped off, forward that to them. And we want your feedback so we can always be improving things. Thanks. And then so now back to questions. Sorry, Alberto.
Yeah, I think we'll just let people continue typing away, and we'll call a close to the out loud portion of today.
I think that sounds great.
Okay.
Thanks all.
Thanks, everyone.
Thank you, everyone. Hope to see you tomorrow.
See you tomorrow. Bye.
Thank you, everybody. Bye.